Hey everybody, good morning from RV Falcor 2. You're watching the dive stream. This is a live camera feed from ROV Sebastian. Thank you all so much for joining the stream this morning, and we just touched bottom for our 731st dive. And what a treat, we've got a little octopus guy sitting here. And in the room with me, I've got Doug and Tyler as our pilots. And behind me, I've got Juliana, one of our marine technicians. And we've also... You want to say hi? Hi. And we've also got an entire room full of scientists, which I hope will introduce themselves throughout the duration of this watch. We've got some SAGE sensor specialists and biologists and microbiologists, geologists. We've really got everybody on this expedition. And with me here in the watch lead seat, I've got none other than Dr. Lisa Levin, who will be leading the narration for this watch. And over to you, Lisa.
Well, hello everybody. Good morning, good afternoon, good evening. Welcome to the El Quisco North Seep site. We've just arrived on the bottom. I guess we'll introduce everybody while we get set up. So I'm Lisa Levin. I am a professor emerita at Scripps Institution of Oceanography at UC San Diego. And I'm out here studying, working with Eulogio Soto to study the animal ecology of the methane seeps and taking a lot of samples for stable isotope analysis to try to understand the food webs. And behind me, we have Pachimunoz. I'll let her introduce herself. She's the co-lead for this watch.
Hola, buenos días o buenas tardes para algunos. Mi nombre es Praxedes Muñoz de la Universidad Católica del Norte y estoy acompañando acá en la sala del Sebastián, observando el fondo frente a el Quisco, en la zona de afloramiento de metano.
Okay, and we have two data loggers, Paula and Erin.
Hi everyone, I am Paula Santiago. I am this watch data logger, so I will be documenting all the really cool critters and organisms that we see during this dive for future analysis. And I am representing Dr. Turber's lab over at University of California, Santa Barbara, where we study microbes on the mud. Hopefully we'll get some really cool ones today on seepage sites. I'll pass it on to Erin.
Hello, I'm Erin from the Geomicrobiology lab of Jeff Marlowe's in Boston University. I'm here to study microbes in the methane seeps and carbonate rocks we find at these really exciting chemosynthetic environments.
Scott?
Sure. I'm Scott Wyman. I'm a PhD student in the MIT and Woods Hole joint PhD program. I'm on board running the SAGE sensor, the in situ methane sensor, from the chemical sensors group of Dr. Anna Michelle at Woods Hole Oceanographic Institution, and collecting some rock samples for my own work on the geology of carbonate rocks at cold seeps and better understanding how they preserve the conditions of anaerobic oxidation of methane through geologic time, giving us a window into how these seeps were behaving in the past in very different climate conditions.
Great. So we met the team. We're landed on a it's probably the base of a very large tubeworm mound. We were here yesterday, and it's going to be the start of our dive. Right now, we're looking at an octopus that could be a granulodone or a moose octopus. And we're also looking at a lot of tubeworms. The hairy aspect that you see on these, it's not really hair. It is a combination are colonial cnidarians, and arborescent agglutinated foraminifera, which are protozoans. And all the hairiness are actually organisms that are probably there using methane. I've worked with my colleagues Victoria Orphan and Shauna Goffretti and Greg Rouse and doing labeling experiments that suggest all these hairy creatures are actually able to take up methane. And we don't know whether other animals are eating them. We also don't know what the octopus does here. We haven't seen octopus brooding here, like has occurred at some diffuse hydrothermal vents recently. There have been new observations, but it's not unusual to find octopus hanging around seeps, I think, because there's so much to eat. All those crabs, for example, and there are a variety of worms and crustaceans that are probably pretty good prey. This octopus probably is more interested in checking us out right now. So our plan for the day is to head north of this site and hit a series of waypoints that were identified by the mappers overnight. We had a night dive on the expedition. The BBC put their cameras up forward and took some amazing imagery of this region and the tubeworm mounds and all the fish in the tubeworms. And then the mappers, Jeff Beeson and Adam Scott, all got to work overnight and identified areas with high methane release and high reflectance that represent hard structures, probably mounds of tubeworms. So we're going to go visit a series of those this morning. But first, I think let's do a little reconnaissance around the big tubeworm mound, make sure all those Congrio that we discovered yesterday are still there. They were there last night. That was great octopus footage. Guess you guys pushed the BBC highlights for that one. Yeah. Oh, we got something back there.
How's the light for you?
It's fine. We'll also be on the lookout for bacterial mats for our microbiology and geochemistry colleagues.
Good to move around. Is everybody happy?
I'm happy. Super happy.
Okay.
We've got our whole dense assemblage in the bottom water of these crustaceans, probably euphausids. They're part of what's called the deep scattering layer, and in the daytime they come down to deep water, like 400 to 500 meters, to avoid predators in the surface. Oop, maybe a cat shark coming our way. Cat sharks often hang out around methane seeps and lay their egg cases on the tubes or the corals nearby. There's another one. That's a rat tail. I was just thinking about the behavior of these fish hanging out backed into the tubeworms, and I think, they're very much like moray eels. And maybe the common name is an eel for these fish. Or Congrio is probably the common name, but I'm not sure if that translates to eel. Do you know?
I've also heard it go by the red
Cusquille?
Yeah.
Yeah. For those people who dive in shallow water, you'll have seen moray eels sort of backed into burrows with their heads sticking out just like this. So the mound's still here, didn't go anywhere overnight. We're looking at a big, beautiful mound of Lamellobrachia tubeworms. These are ciboglinid polychaetes that are really special because they don't eat in the classic sense. They have no mouth, no gut, no anus, but they have a large organ called a trophosome that's full of symbiotic bacteria that can oxidize sulfide. So they're all clustered here, obtaining hydrogen sulfide, which was probably hydrogen sulfide, which was created when the methane seeps up and is acted on by the anaerobic methane oxidizing consortia. Ooh, we got a bubble, or two or three. That's good. These are methane bubbles originating from, most likely, from the decay of organic matter deep inside the crust. They're not deep, but down below the surface of the mud. So there appear to be two forms of these worms, long, elongate ones and sort of curly, gnarly, clean ones. And Greg Rouse, who's on board from Scripps, is going to be figuring out whether these represent two distinct species or not, tubeworms. Tubeworms are an important part of the seep ecosystem because, as you can see, they form really large structures. And these structures provide habitat for a host of other animals. I was just this morning in the lab looking through a clump of hydroids that had been pulled off this mound, and we found, I saw isopods and amphipods and a few other weird protozoans that I didn't recognize. There are small crabs that hang out on the hydroids and on the tubes, as well as the big ones you see with the cameras. So not only do they provide substrate, they're clearly providing some refuge maybe for predators. In the case of the fish, maybe some refuge from fishermen. It's a little hard to tell. But then this big edifice, which I think we measured yesterday at something like 7 meters.
Wow.
Is that right? It also alters the currents in the region. It can intensify flow, and so other things that are filter feeders might benefit from being on and around and above this structure. It looks like a closed up anemone, maybe. Again, the hair here, these hairy tubeworms, are covered with hydroids, colonial cnidarians with stinging cells, but they may also have the symbionts with the ability to oxidize methane. We're still investigating that aspect. One of the really interesting things that showed up last night on the BBC footage was that these fish had, at least one of the fish had one of these spider crabs sitting on its back cleaning off parasites. So maybe that happens at night when usually fish are more active in the daytime and invertebrates are more active in the nighttime. If you put out a camera, that's usually what happens. I'm not sure about the cusk eels and when their biggest activity period is, when they come out and feed. Yeah, I don't know, for some reason, it just struck me that they look just like morays, sitting there poking, like in coral reefs and things, poking their heads out of rocks. We did a lot of sampling at this site yesterday. We took suction samples and we collected a few very specific starfish and snails and so on. So we probably won't disturb the mound today, and we will be heading to Waypoint 2 after we've had a good look here. Enjoy the scenery.
Were we able to get any steam level until the month yesterday?
Oh,
Or should we-
maybe that's a good point. I think, Erin, the bottle failed, right? Should we get a Niskin here?
I don't need it anymore. The incubation's already set up.
Maybe I'll take one. Because I didn't take a sample yesterday, so I'd like to drip a Niskin, probably just over the top,
Okay.
please. Ooh, I'll probably get some euphausids. Look at them all. It's pretty cool. One of the really interesting things about these vertical migrators is a lot of demersal fish will actually eat them when they migrate down to the bottom. When they hit the bottom, they can't go any further down, so they stop and hang out like this. And we find them in the guts of bottom fish, especially in places with a lot of oxygen.
Okay.
So that's Niskin 1.
Let's see. And then just crank up crank this. Okay. Yeah. And open this a bit. Whoa. All right, going for Niskin 1.
Reset.
What's that?
Reset it.
Oh, yeah, put it to 30 days.
For those on the chat, we're starting at the same mound we started at yesterday.
Niskin 1. should
We're
be
at the
fine.
northern end of El Quisco,
Sit.
so Niskin 1 is taken.
Yep, Niskin 1.
And
I can see
it's-
the bottom. I can't see the top, but I can see the bottom.
Yeah, that's tie wrap.
Yes, that's right.
You can tell it's closed, okay.
And you can see where I was at, just right over the top of the mound there.
Yep, great.
That's how you can fix the cameras.
Camera back.
Thank you.
All right.
How, can we zoom in on this set of facets? And I don't know if any of them will show up more clearly or what happens when
try.
you zoom in.
These the small ones?
Well, they're more dense atop the mound, it seems like.
Okay.
just to try to,
Just the lowest because
we don't often get a look at the diurnal vertically migrating fauna. They're also called the deep scattering layer. Some of these are resident, but many of them move up and down each day. They are a really important food source for pretty much all the higher order consumers in the water, and also they're a really important part of the biological carbon pump, moving carbon from surface waters into deep waters and helping to sequester it. So every so often, a Euphousine swims into perfect field of view at the camera. We can see them. Oop, there's some maybe small rat tails. Okay, that's good. Just curious what we see if we zoom in. We can zoom back out. Would be really interesting to know if these fish have territories and they each have a specific part of the bush, the big tubeworm mound that they hang out in, and they always go back to the same spot. So we're starting at the same waypoint 1 that we had yesterday, but instead of going to the east and following our targets, we're going to be heading to a handful of targets to the north. And this whole site is north of the El Quisco region we explored a couple of days ago, but it's about 1 kilometer further north. There's a skate. I think it's a skate. Beautiful. They also lay egg cases at seeps and vents. It's a lot of fish biomass in here. These are really big fish. They're harvested commercially in Chile, and they taste great for those people who eat fish. Understanding how commercially valuable species is associated with methane seeps is really, really important for management. I could see at some point that this area would be proposed as a marine protected area or as essential fish habitat. I think that this is one of the most interesting things we've seen about these tubeworms is their use by the fishes. It's really interesting how some parts of this mound have very clean tubeworms that aren't colonized by hydroids, and much of the mound is covered with hydroids. What causes the clean parts to be clean, whether they're young tubeworms or a different species that has a chemical that protects them, we don't know. These mounds are much bigger than any lamellobrachium mounds I've seen elsewhere. We studied them off Costa Rica. We had one bush called the Volkswagen that had on the order of 13 or 15,000 tubeworms in it, but I think we have many, many more here. You can see they're sitting atop carbonate rock. That's the rock we see in the view now, and that rock is made by microbes. Would you like to speak Pachi, about what we're seeing? Spanish?
Como decía Elisa recién, estas rocas son generadas por los mismos microorganismos que viven en los sedimentos, hablando de una forma más general. Específicamente se llaman carbonatos autigénicos o minerales autigénicos que se forman in situ. Algunos de ellos por la oxidación anaeróbica del metano. Acabamos de detectar en esta zona un flujo importante de metano a los que se asocian varios microorganismos. Mi especialidad es la geoquímica, así que vamos a ver varias cosas interesantes en estos testigos.
It's a big anemone. A different one. Another cat shark. They look pretty small compared to some of the other creatures like the anemones. It's a different crab, a Paralomus-like crab down there. Where's the pointer? Could you zoom in on this just so I could see what it is, please? Looks like a sea cucumber. Oh, it's an asteroid, maybe. Let's see.
Figures.
Well, it's okay. I was just trying to figure out if it was a sea star or a sea cucumber. All right, let's go ahead and head over the bottom to Waypoint 2. If we see any really large bacterial mats, we may stop the core. This is such a beautiful mound of tubeworms, and we are so lucky to have such great people and equipment to be able to view this. I don't think anybody prior to this expedition had ever seen this feature. It's really interesting. We're not very far offshore, I think 15 kilometers or something like that, but this is very typical of the deep ocean, that right in our own backyard are amazing, still to be discovered features In many cases, they've already been damaged by fishing gear or maybe oil extraction or oil spills, but often before we have a chance to discover them. So we're headed.
Go ahead. Hey Tony, good morning. I got a new waypoint for you.
Okay, Lisa, your audio's back.
Sorry?
Your audio's back.
Okay. Oh, so it wasn't, where'd it go?
No, I just muted it.
Okay. So the thing I said about the hydroids didn't,
Yes, please repeat that.
I could repeat it. Okay, well, all right. Well, these purple little tree-like creatures are actually probably Heteropolypus, which is a type of cnidarian, maybe a panaculid, and used to be called Anthomastus. And then this really slim single polyps that we've been seeing, also purplish, are hydroid polyps. The shrimp, the little shrimp are really interesting out here. They cover the bottom. It'd be kind of cool to do a nearest neighbor analysis and see if they've got themselves all uniformly spaced out to cover little territories or not. Sometimes predatory animals like this are territorial. They look pretty evenly spaced to me. So we're headed to Waypoint 2, which should be another large mound feature. How many meters?
Point 2 is, from our current position, 165 meters.
Okay. So while we're headed there, I'm going to let everybody in the room talk about the science that they're doing. And let's start with Pachi. You want to talk about what you do when you take sediment samples?
Ok. Bueno, estamos volviendo a algunos puntos que fueron mostrados anteriormente para obtener muestras de sedimentos con un sistema que se denomina push core. La idea es hacer la geoquímica de estas zonas y asociarlas también con los organismos en cada estrato sedimentario para poder entender cómo viven los microorganismos y también macrofauna y organismos más grandes. Entender la productividad impresionante que se ha observado en esta zona, considerando también que asociamos los gases como metano y ácido sulfídrico con elementos más bien tóxicos, y se está viendo acá que hay una gran cantidad de organismos que se asocian a vivir en estos ambientes con bajo oxígeno y con concentraciones inusuales de gases como el metano.
Paula, you want to talk about the samples you're taking?
Let's try this in Spanish. So, ahora es interesante porque toda mi vida he hablado español, soy de Puerto Rico, pero la ciencia, mi preparación graduada es en inglés, así que me encantaría explicar lo que hago en español. Sí, yo acompaño junto a los sedimentos que Pachi recolecta, yo tomo un poco de esa muestra y la analizo la composición microbial. Eso lo hacemos mediante el análisis de ADN, ARN, y lo que nos interesa saber es quién está ahí, quién está consumiendo o produciendo el metano, si estamos cerca de una ventana de metano. Por otra parte, también las muestras que tomamos de la macrofauna, así que usualmente los organismos que vemos como los camarones, las estrellitas de mar, a esa yo estoy tomando un poco de su tejido y ahí le realizamos análisis de ácidos grasos, porque es bien interesante cómo los ácidos grasos de la bacteria que produce, que consumen el metano, no cambia mucho entre eso y cuando un organismo de un alto nivel trófico la consume. Así que animales más altos en la cadena trófica, como pues, las estrellas de mar, los erizos, como lo hemos recolectado, en teoría no van a cambiar y nos van a poder demostrar si están beneficiándose de la ventana de metano o no. I'll pass it down to Erin now.
Yeah, so I'm interested in the microbes living in the sediment and the rocks at these cold seeps. To study them and figure out who's there and what they're doing, we do a combination of DNA analysis, geochemical analyses, and then bridging the two, we have some incubation experiments set up where we'll be able to track which cells are active and which are not, and then track how much methane they're consuming over time. And that'll give us a sense of the cellular activity at these sites.
And what I've been doing the last few days, collecting carbonate rocks, and we actually take all the animals off. We let them crawl out overnight because many have worms and other crustaceans, other animals that live deep inside the crevices, and then we determine the surface area of the rock by wrapping it in a monolayer of tin foil and weighing the foil, and then knowing how much a centimeter squared weighs. So we can calculate the density of animals on the rocks and the composition and the diversity and so on. We are interested, carbonate rocks are very rich in fauna and important habitat for the seep community. We also take tissue samples for stable isotope analyses. Delta C13 and Delta N15 bulk analyses can tell us a little bit about whether the carbon and nitrogen sources are chemosynthetic in nature or whether these animals are eating food that falls down from the surface that was fixed photosynthetically. There's our friend, the big shrimp with the long tentacles. I'm not sure Being an ecologist, I'm pretty bad with names of things, remembering them. But we have clearly two types of shrimp here, really, the big ones and the little ones. Seeing more big ones here. I'm sure the bigger ones are harvested commercially. The small ones might be as well. Possibly with traps. The sediment is not flat. It's full of mounds and burrows, and we haven't really seen who's been making these features. They're often made by burrowing shrimp, sometimes calcium acid shrimp. You can find them in intertidal mudflats, shallow sand reefs. But turns out there is a group, the axinid shrimps, that live in chemosynthetic environments and will make these types of burrows. So it would be really interesting to know who's down there, but we don't have the equipment or even the personnel right now to investigate that. Hachi, there weren't any shrimp in the multicore, were there? When you didn't get a burrow? No. Yeah.
We didn't see.
There's a little cluster of rat tails. Don't know which rat grenadier species this is. I know there's a small one called Nizumia. But it might be Coelorhynchus. I think that's the name Maritza gave us the other day. So the number of fish have picked up. We have to ask what's happening here, that there are more of them. Are we getting close to the mound, maybe? Cat shark, Heteropolypus is the anemone-looking thing, the big red one. It's not an anemone, but it's related. Ooh, an acroac shell, maybe. When you see a dark structure with white in the center, clam shells, those are solomyid clams. What is Puedes venir? Somebody can, no. I see, that's something else. So typically, if we get near a structure, we'll start to see clam shells, sometimes higher densities of the background fauna even will cluster around a tubeworm mound. Ooh, bacteria. Could be comment on that a little bit? See if it is bacteria. Might not be bacteria, it might be a coral. Let's see. Yeah, that looks like bacterial mat. Small bacterial mat. When we see bacteria at the surface, it tells us that there's methane seeping from below and hydrogen sulfide present, because these are mostly sulfide oxidizing bacteria. There's often some methanotrophs tucked in there as well. We see between the mat and some scattered clamshells, it suggests we're getting closer to an area of active seepage. Tell us about bacterial mats, Erin. What do we know about them?
Yeah, so bacterial mats, like Lisa said, are an indication of a really rich chemical environment that the microbes can really feed off of. You know, I think a lot of times when people think of microbes, they maybe don't know what to picture. They are, by definition, typically invisible to the eye. But there are some really special species like Thiomargarita, or more likely here, Biota, that are capable of creating these like long filamentous structures that we see here. We found some really beautiful ones so far here at El Quisco.
So we should be coming up on something. But of course, won't always have a giant tubeworm mound. Can you tell in the sonar?
It should be straight ahead of us from here. There's something, there's a shadow, so that means there's something sticking out.
Oh, here we go. I see tubeworms ahead.
Plastic cups.
And a series of plastic cups, like somebody's marking the way.
This is probably your target at Waypoint 2, right?
Yep. Let's check it out, see what's here. Looks like tubeworm clusters on some relatively flat carbonates. We have the usual cadre of hydroids and small red corals that are probably Swiftias, and maybe some bryozoans, stalked bryozoans. Looks like a crinoid attached. Some sponges. They look a little different. Lots of clamshells. It's been hard to find big clusters of living clams here for whatever reason. This region is probably all has diffuse methane seepage, or the tubeworms wouldn't be living here. So this is a relatively low relief mound, maybe 2 meters high, 1 or 2
Maybe
meters.
roughly that, yeah.
But it's kind of broad around. More plastic cup debris. Plastic doesn't decay very quickly down here. Crinoids again.
Right. I'll have to go back the other way to catch the rest of it. So I'll put a turn in my tether.
Sorry?
I'm gonna come back around this way to catch the other side.
Okay.
So I don't get a turn in my tether. Squid
Oop, a squid.
came down for a snack.
Skate. So after we sort of circle it all, well, I'd like to just come in close to some of the rockier part areas, and we'll do some imaging of the fauna. Maybe a suction sample. There's a big Gorgonocephalus type basket star on the rock over there. So this side looks a little rockier. With steeper slope. Could
Pretty
we
much
zoom
all the
in
way around
on
it.
the basket star?
Sure.
Just to get a beauty shot, please. So these are actually brittle stars. They're closely related to disturbance, so we can back off if we need to. There's a couple of crabs there. As far as we know, these creatures aren't associated with symbionts. They're up high like this to filter feed. But of course, I don't know if anybody's ever looked for symbionts, and these echinoderms generally don't have symbiotic bacteria. It's perched fairly precariously at the top of a tubeworm. Not sure what it's hanging on to. Little white polyps beneath. The basket star are zooids of some sort, cnidarian, zoanthids maybe. And there's some agglutinated arborescent foraminifera attached to them.
So what are the hairy bits on this crab's arms? Is it similar to that of yeti crabs, or?
They're apparently spines, and those spines could host bacteria. Yeti crabs that Erin's asking about are the dancing furry crabs from Costa Rica and from Easter Island at hydrothermal vents, and Costa Rica at methane seeps. But that's a good point, that these crabs have at least the structure for farming bacteria on their arms. Maybe we should try to see if Greg's up. Maybe he can tell us if he's already collected these or not. Could we zoom back down to the hairy crab underneath, please? Hairy armed crab, I should say. We zoom in. So Erin's asking about the spines on these crabs, and they're definitely distinctive and potentially could be used for hosting bacteria, which are then eaten, harvested and eaten. Just send a message to Greg. How hard would it be to suction that crab up without disturbing the basket star?
Don't know. Depends on how strong that tubeworm is
Yeah.
and how much that crab's holding on.
Well, we could look around for another one also. I'm sure where there's one, there's going to be others. There's quite a diversity of Galathea crabs here.
Just tell me when you want to move on.
Why don't we back off and just look and see if we see any more of those crabs with the spiny claws. Might be useful to have collect one. So most of them don't.
It's not the same, is it?
I don't think it's the same one, but this crab also has a lot of spines on its claws. The animals at hydrothermal vents and methane seeps have come up with really ingenious ways of farming bacteria, hosting bacteria, associating with them so that they can use them as food. So that one doesn't really, it doesn't have the same features. There's a hermit crab. No, it's a different one, I think. There's probably at least three or four different crab species here. Well, that one does that look more like it? Yeah. I know we got one yesterday. That one could be, maybe we could try suctioning that crab. So this one's not covered with mud. It's got a lot of really fine spines on its arm, on its claws. And it looks like it's sort of upside down. But I'm worried about disturbing the basket star, so I think we'll leave it.
Okay.
So you can see this crab looks very muddy, but in fact is probably got either mud on its spines or bacteria on its spines, on its legs. Why don't we go ahead and take a suction sample of this, if you can reach it.
No, I'll have to move first.
Of the one on not the one under the
Right.
turtle star, right? Just to be sure.
What jar do you want it in?
5, 6, 7, or 8.
All right, we're lined up with 5 now.
Okay.
All right, Tyler. Let's see if we can find it.
Let's get this little muddy guy.
I don't see him anywhere.
Where'd he go?
It's not that guy, right?
Well, that guy's pretty muddy
Whoa,
too.
that's very muddy one.
We could try that one also.
Oh, jeez, look at this.
Let's-
Wearing a ghillie suit.
Yeah, let's try. Well, will they fit? They probably won't fit in there, will they? It's the latest style. Mud fur coat.
Where did that come from?
Yeah, so the bigger fat brachyurin, let's get one of the ones we were just looking at.
Okay.
Greg said we don't have one of those. Or if he gets
See him?
stuck at the mouth, we could
See him right there?
drop him
Yeah.
into a coral tube or a bio box.
I know when you're ready.
Ready?
Suction.
See it moving still.
Yeah.
Oh, he's still down there.
Yeah. Okay. See if we got it.
Did we get him?
Not sure. Looks like he's hiding out down there.
Yeah, he's tucked under the worms.
You see the other one that was walking around? Ah, there. See him straight ahead of your nozzle? Right there.
Camouflage.
Yes, good camo.
Nice spot, Doug.
Still got suction. He's moving.
Let me see it on my shit.
There you go. Just about. You're over the top of him. There he goes. There he goes.
Great. So there's a really big diversity of crab types, Galatheid and brachyurin crabs here. We're hoping to at least get one or two of each type so they can, some of these will probably be new species if they're seep associated. Do we see the other hairy type?
I'll have a look for him.
So in the chat, we have had a request for people to talk about their career paths and how they got interested. But I'm thinking the other Galatheid that we were looking at
Oh.
originally.
Is that?
Right there.
I don't
See it?
think, that one, yes. This guy.
Crabs are hiding from you today.
They're really well hidden, aren't
Yes,
they?
they are.
It's funny, they shouldn't need camouflage, it's dark down there, so.
Right.
I don't think there's been enough science pressure to shape evolution.
No. Still got suction.
So Erin, you want to talk a little bit about how you got into science and got where you are? How you
Sure.
got interested?
Sure. So I'm still a student myself, but I'll still give you a little background on how I got here. I was actually really interested in astrobiology, and also how life functions at the chemical level. I knew I loved chemistry, but I wanted to put it in the context of environmental microbiology. because to me, chemists are, nature is just naturally, just natural tiny little chemists, especially ones at chemosynthetic environments. And so I was trying to figure out how to get into astrobiology, and my microbiology professor at the time pointed out to me that deep sea biology and astrobiology actually have a lot of overlap, particularly in the context of like ocean worlds, like on Europa or Enceladus. And so I joined a lab at my undergraduate university, University of Rhode Island, that was doing microbiology work in the deep sea and along the coast, looking at these cool chemosynthetic endosymbionts. And working in that lab, I really started to realize my heart was really with the deep sea, and I was just more drawn to the ocean. It felt more pertinent given its relation to climate change and conservation. I really enjoyed the fieldwork aspect of it that I was missing in astrobiology work. So I really shifted my focus there. After graduating, I spent a year working at Woods Hole, and along the way, got some seagoing experience. Grad school, because I had my own research questions that I wanted answers to, and found a lab that was a really great fit. Pella, do you want to talk about your path?
Yeah, of course. Thanks, Erin. Yeah, I am a student also, same as Erin, a graduate student, and I started off undergrad wanting to go to medicine school. So a straightforward path to that is doing an undergrad in biology work, and that's how I started. And I always try to tell this funny story, but this is not precisely why I got into deep sea research. But I had to wake up for shadowing one day for cardiologist, and when I woke up, I realized that this is not one. I do not want, this does not give me energy to wake up in the morning, so I don't see myself doing this for the rest of my life if it does not give me joy to wake up in the morning. And after that, I was just trying to find any opportunity within the biology, because I knew I loved biology, to explore and do research. And from there, I noticed that I wanted to get research experience, so my first research experience was within doing river work and studying its microfauna. And from there, they were asking for volunteer divers, and so I got my diving certification to go help with the coral nurseries, because in Puerto Rico, the corals have a lot of value in the both culture and as a protection for our coast. So they're very important, and we work a lot in. So that was my first experience with ocean sciences, and from there I hadn't looked back. That was about 6-7 years ago, and so I was a volunteer diver, and then that became into a job, and it came to the point that I really wanted to study into the frontier of science, and for me that was polar work, and that's how I got into internships with NOAA and Ocean Exploration Trust and the research vessel Okeanos Explorer. And when I noticed that the deep sea was also a frontier in science, I was amazed by it. And from there, I have always tried to look for opportunities where I could get involved into deep sea research. And yeah, I think that sums it up. After a while, I really wanted to apply my research skills back home in Puerto Rico. So I developed an internship back on the island for undergrad, so they could experience the shallow water research. And for me, long term, I wanted to bring all of this back as well and bring the frontier of science back at home in the deep sea. Pachi, ¿cómo llegaste a estudiar ciencias marinas?
No, larga historia, muchos años ya, demasiado.
Sabía que quería esto. Sí. Oh, qué bonito. Fue fácil estando en Chile, tenía los recursos para poder estudiar fondo marino.
No.
No, nunca, no siempre fue así.
Diría que muchos de los que estudiamos en mi generación es porque veíamos los programas de Jacques Cousteau.
Oh, wow.
Eso es.
El vinito. Lisa, were you also inspired by Jacques Cousteau to get into
No.
deep sea research?
I had a great deep sea biology teacher when I was a graduate student named Bob Hessler. He's no longer alive, but I was a student at the time, hydrothermal vents and seeps. Well, vents were discovered. Seeps weren't discovered until after I was a professor. But I started off doing shallow water research for my PhD on polychaetes and mudflats, and but was always fascinated with the deep sea, so kept one foot in the deep sea, collaborating with Craig Smith on some research while I was a grad student, even though it wasn't my PhD. And over the years, I've become more and more fascinated with the deep sea and also its link to climate change and to humans. But I credit my initial interest with great biology teachers in middle school and high school and college who helped encourage me to go on into marine science.
Is there anything specific you're looking to capture here, Lisa?
Now we're trying to get the crab through.
Yeah.
Okay, so just flushing it out.
But I just figured in case
There's the other one.
we figure out.
Yep.
Oh, well, you could try for that one. Maybe one would push the other.
Maybe.
It's tough.
Yeah.
It's in there now.
Well, it's in.
It's another fish head down here. Sonny. Doing your favorite job. Section 6. So good.
Waiting for some crabs to come down. Did you get them or no?
I got it, yeah.
You did?
Yeah, it went
Oh, yeah,
in.
you sure did.
Did you look in the nozzles and stuff?
It's a crab log jam.
Crab jam.
Oh.
That's not gonna work.
So this site has a lot of bryozoans. Maybe more than some of the others.
I don't see it in there.
No, not in the.
Not in the immediate. Let's see what drops
So we've
down.
sectioned 2 of these interesting brachyuran crabs with a lot of spines and what looks like hair that made out of mud. And they may be stuck in the suction hose.
Kicks out.
Might just be a matter of time before he comes down. All I got on is, block.
There's a jellyfish hanging out near the bottom. That's really interesting.
Pretty sure I came to the right. But if it counts up, go the other way. Sure.
Go to 0.
Thank you.
What else is on it? Also, we didn't touch.
The plankton is interesting. It looks like perhaps our lights are attracting arrowworms, squid,
Just watching their crab.
jellyfish.
Oh, he's
Yeah.
little.
That nice squid.
That's my birthday song.
Just get it jammed in there.
Just have it push the crabs through.
Yeah, that thing will push the crabs, that's for sure.
We don't think very much about the fact that the seep could be feeding the plankton, but it's not unusual to have pelagic animals come down and feed near the bottom or at the bottom.
That's awesome.
And as you can see, there's a lot of euphausids and small plankters hanging out, maybe mysids hanging out near the bottom that could provide food for these guys. Okay, well, we're not sure if there's crabs in the suction hose or not. But I think we'll continue to move around this waypoint and maybe head to waypoint 3. Nobody needs a Niskin from here, do they? Do you need anything? We haven't seen any bacterial mats big enough to core.
I think it's better than these bottle
What?
inside. These bottles are better inside where we
Core.
take those ones.
Yeah. So we could call this the hairy crab site, since we saw a couple of species of spiny crabs that look hairy. So this site has skate and catfish, cat sharks, sorry, not catfish. Some large whelks. So this is just like 30 or 40 meters away.
Does he have bird traps on? Usually runs bird traps.
So there's some scattered carbonates here with the usual suspects, corals and hydroids. There's another bacterial mat. So we're getting again towards some more seepage. Some sporadic carbonate. Debris, lots of that.
Yes, I'm sorry, that's all.
And then tubeworm clusters sort of surrounding a low carbonate mound. Another basket star. Can we zoom in on the basket star just again to take a quick look at that?
Yeah, you do.
They're really big. I wonder what fish that is hanging out underneath. Tubeworms. So beautiful. It's always sort of precariously perched at the end of a tubeworm to elevate itself up off the bottom. As I mentioned earlier, these are basically a form of ophiuroid or brittle star, but with many, many arms, branches on its arms. You guys got a picture? Yep. Okay. So there's another potential Congrio under there. It's one of the first ones we've seen hanging out under tubeworm's knot on that giant mound at Waypoint 1. So waypoint 2 is I'm sorry, we're at waypoint 3 now. It is a low carbonate mound, sort of like at waypoint 2. So if once we get out of this, we'll probably want to come back around and circle and take a closer look at it, just see if there's anything new and different. These might be black coral cinderus. To see if Fauna wants them. Can we just kind of come around, back around this feature? Another Humboldt squid. We were noticing that the squid and the Congrio had sort of different distributions. When we saw a lot of squid, we didn't see the big cusc, and when we saw a lot of cusc, we didn't see many squid. Oop, octopus maybe over to the right. It's pretty unusual flattened behavior there. Probably trying to sleep. Oh, we've woken him up. Sorry. Looks like an old guy. Octopus are quite intelligent creatures. There's been some great novels written about them recently. Okay, so let's try to go back and survey that mound feature we just went over a little bit more. I thought I saw some black corals that we just went over, but if we see them again, we might want to sample. Is this fishing line on the left? Right there? And a burlap bag or some kind of sack. So marine debris. Guys.
Can you zoom in on that?
Does that look like fishing line?
No.
No?
No.
Looks biological? I don't know, there's a bag right there.
Debris, right in the middle there.
But I don't know what else these would be. Maybe there's something. I mean, it's definitely colonized by creatures, by hydroids and forams. Could you zoom over or pan over to that line? I just want to look at it more carefully and see if it's an organism or rope that's,
Oh, look at crab.
Oh, look, there's a crab.
Yeah.
The red eyes.
See it?
but
Is this the same pump?
Yeah.
they're so well camouflaged, you can barely see them. So we're looking at a crab that's sort of buried there in the line. Full of spines.
Almost.
I can't tell.
I think it's close.
So what I don't know is whether these linear things are rope or biology. And whether there's certainly some fishing debris down there. Some kind of sack. So we have discolored sediment, black sediment. There might have been something organic there that was in the sack that decayed. There's also some interesting may pan over to the right a little bit, please. I'm interested in what those are. Serpulid polychaetes on the worm tube. Maybe they're polychaetes. Are we as zoomed in as we could be?
And remove that a little. Zoom
You wanna get closer?
in.
Can you
Maybe
get
our-
it? We might want to collect that tube. It looks like it's got something different on it. Could be a Nemertian all wrapped around there. Yep. I don't know that it's a polychaete unless those are calcareous tubes.
Okay, zoom in.
Like sparkly like the snow, you can see. Or like, what is it in this sparkly?
I'll show you what it is.
Or, that would be awesome.
So it's like shimmering.
Yeah. Oh, like the pixels of
Yeah.
mission thing
Yeah.
versus if it was like here.
See now.
Oh, it's
there
sparkling
it's-
further back.
Yeah, because the focus is a little better.
So right there is good.
Yeah.
That is amazing.
So you guys got a picture of it? Do you think we could collect the tube and put it in a bio box?
Yep.
Or in a coral quiver, whichever would be easier.
Well, yeah, I'll open a quiver, but if it comes out too big to fit in a quiver, we'll put it in my box.
Okay.
Starboard round coming out.
Yeah, let me get that. I'm in the way right now, so I'll show you the camera.
You can probably reach one of the front ones right there.
Yeah. I need you to come out and this back on. Look at you.
It needs to be on this side, yeah.
I guess like,
Yeah.
oh, there's one up. But you can see, just to get rid of.
I hope we can collect the tube without pulling out the whole clump. See.
This side of my arms on the back.
Probably.
Just leave it. Yeah. Let me just grab that. It's all the way up there.
I'm good enough. Yeah.
You think so? Yeah, I'll find more. Is it all about there?
No, I came out.
Let's go on. Yeah.
Yeah.
Greg said maybe those are egg masses, but we don't know whose egg they could be, lining the tube like that. Gastropod, probably.
Are all the coral fibers?
Yeah, I think so. You haven't put anything in any of the coral fibers yet, right? Coral fibers are empty. Yeah. Good for that.
I guess we can see it.
One is the tail of the, yeah.
I just can't see it. I will make on the camera.
Remember, it wants a little tail, right?
Yep. Should be fine enough. Ooh, okay. Maybe if you wouldn't mind coming in just a little bit on this.
I think Greg's probably right, that we're looking at little egg strings that many gastropod species will lay their eggs on hard substrate. Still think that we're right next to that mass's fishing line on the left of us. Little small line.
And just grab it above the root and cut it.
Do you want the worm intact, the tube intact?
No, it doesn't have to be.
I'll see, now they have a different angle, I'll try pulling it out to the right. If that doesn't work, I'll cut it.
But we want to leave the stringy part intact,
Right, the white
the white,
tetra mass.
yeah.
Just want- outside if it doesn't come, definitely snip it.
Okay. Take the drug.
Alright, looks good. Should turn the lid
Yeah, rotate the drug. So the lid is on outside.
Well, no, because if the lid's on our side, then we can see the lid better, but then it won't, we grab it on
Okay.
with fingers. Another one, don't want.
Ooh, you cut one of them. Got it, great. Might just be easiest in the bio box, don't you think? Or, I don't know, your call. How, if you think it'll fit in the coral quiver, it's fine. It's a good example, if these are cases, of how the tubeworm bushes serve so many functions and could be a gastropod nursery function here.
Just don't bring too far in because we'll come out of the light, so, but yeah, I can definitely come a ways. When we come up a little bit, just make sure you have a dark background.
Somebody on the chat's asking about studying the growth rate on tubes, of the tubes and the tubeworms, and this has been done with a dye where in the Gulf of Mexico, where I think the submersible Alvin probably came down and enclosed some tubeworms with a green dye that then marked the growth edge. And they came back a year or two later, maybe 3, I forget. Came up with estimates that the tubeworms were 300 years old based on the amount of growth since the mark. It's a great, great shot. Thank you. Another idea here from Greg is that we're looking at baby tubeworms in some weird form on the adult, on another tube, but we'll see what it is when we collect it, bring it up.
I'm sure I can remember it looks pretty small.
Tubeworms are thought to live hundreds of years and grow slowly, but in some settings, like in the East Pacific Rise And even on the Wanda Fuca Ridge, they can grow quite quickly at hydrothermal vents. They're less well constrained at seeps, I think, in terms of knowing the ages.
I was just checking my set. That handle's also not.
There you go.
It's stuck to the
Great, thank you.
Just needs a- There you go.
Is it? Or is it still right there? Another set. I think that's
There you
it.
go.
So, Coral Quiver one. I'm just calling it a tube with eggs, question mark. But we don't really know what's on it.
Lisa, what would be the next steps so we can know what type of egg case is it, if it is an egg case?
Well, it could, if it is an egg case, or even if it isn't, it could be sequenced. And the molecular information would tell us which phylum it's in. And then if it matches with any known creatures, gastropods, or if it's a baby tubeworm, it would tell us that probably. Greg's lab would do that. But we can also take a look at it, probably tell something up under the microscope. Thank you. Okay, let's continue surveying this site. We'll just continue to drive around it with looking closely at the bottom and see if we see anything else unusual or interesting. For a little while, we had jellyfish and squid right above us. Is that a squid out there?
Yeah. See the little lights on them though?
The lobes?
Yeah.
Just ended up seeing more snow.
Oh, yeah. Alright, then another.
I can brighten it up with the ISO too. Is that better? Autofocus is doing weird stuff.
Yeah, that's good.
Does anybody need background cores? One. You need one. Well, we're at a seep site, so it's not a good background core site, but we aren't far from background.
It's me. Oh, alright, right.
And we
Patchy
have
also
some-
requested one, so I can share one core with Patchy on a background.
Okay.
Just like adjust a little bit because
They're in this bacterial mat's not, oh, she's not here. I think is this the same bacterial mat we saw on the way in?
Yeah.
Probably.
It is what it is.
Two in a room again, right? And the two encumbered lock again. The
Lisa, Erin is back for the voice.
Okay, well, you can ask her if that bacterial mat was big enough, but I think the answer is no. So if we could stop and just zoom in on the rock and see if we see any other creatures that we haven't seen before.
Yeah, do your zoom in.
This looks like a black coral right here. I don't know if Anna would want this. Do we know if Anna is around? Do you think Anna wants the coral?
We'll try next time.
Can we text her? Just have her take a quick look at it. Looks like something with the name Pathes, like a Bathypathes or a something Pathes. So over to the left, we have encrusting bryozoans on the rock and a variety of anemone types. Coral has a crab.
Is the substrate here carbonate rock?
Yes,
Thank
most
you.
likely. A little piece.
Yeah, but I think it's a little bit hard.
Well, we could take the whole coral. It's small.
This is
It's
small?
like, it's probably
it's
like this
small-
big.
Okay, it's small, okay.
I don't know, about 20 centimeters.
Do you think it's 20?
Because
Lasers
the
are in
two
it.
lasers there.
No.
20, it's like this.
Is that too
Okay,
big?
let's try a piece, otherwise.
There's a crab on the back side of it, I think. But that's okay.
Are you going to collect the stone?
No.
A small piece of it. Just coral.
There's also a nice hairy crab on the bottom of it.
But it's art.
These are the kinds we were just collecting.
Yeah.
There's one.
I don't know if we'll be able to cut it, we might make a mess of it.
Okay, okay, collect the entire and the rest of the business go to the doctor. We stay in Chile.
If we can, do you want the basil plate?
You want it?
The basil plate, just, not, okay, well, like if we're taking the whole thing.
If he's taking the whole thing, do you need the base?
If you can,
Yeah,
yes.
possible. Okay.
Depends.
Yeah, we'll look at it and see what it,
Because if you take gold, probably take them first.
I mean, we can collect the rock. If it's easiest to just pick up the rock and put it in a bio box, we can do that. Then you have the coral.
yeah, just poke at it and see
For
what.
you it's fine,
see
the rock?
what-
For you it's fine, the rock?
I'm not gonna take any more rocks today, so. That's what we decided, we would collect them if there were the way to get an animal.
Sorry, no.
But we've
Yeah, let's see.
We've processed 15 rocks already from El Quisco, so that's probably enough. We'll see. But yes, feel free to pick up the whole thing and put it in a bio box, so it'd be easiest. And we get a sponge in the bargain.
Lisa, I have a bryozoan question for you when you.
Oh, yeah.
We'll probably have a crab.
Are these encrusting bryozoans the same kind or same variety that we might see elsewhere? Are they relatively ubiquitous, or do they have different species? I know I've certainly seen them a lot in like kelp in like the North
Yeah.
Atlantic.
Well, they're probably related, but I'm not enough of a bryozoan systematist to know.
Nice.
That looks nice.
I rotated to see if we could get a picture with the plate.
Crab will have to find somewhere else to hide.
So I'm gonna try to lift it where we have it dark.
Yeah.
And drop it.
Picture is quick.
It's good.
There we go.
Nice.
Thank you for the close-up.
We've got an associate on that lady on the back.
Yeah. It's a lot of stuff.
Would we want also any skin
Alright,
bottles
just
around
stick
this
with
area?
me.
I'll wait till you're
After?
ready with the mouth.
Yeah.
Oh, we need to ask Anna that. I'm gonna guess maybe yes, but
Turn the light back on.
See if the lowers are gonna play those.
Yeah.
this particular part of the carbonate at this site has quite a lot of corals, but no tubeworms. And so the interpretation here would be that it's, if it is a seep, it's a late stage successional seep. There's some tubeworms down at the base, but mostly there's probably not a lot of methane here. When this, yeah, it's okay.
One has
No, we have, number one's already taken, so it'll be number two. Anna, can you stay in and just look at some of the other corals
Yeah,
here?
stay here.
Because this site has quite a lot of black coral, different
We're
sorts, I
doing
think.
it for maybe for
Which one do you think? Yeah, I'll
You're
reach
going
out.
for 4A?
When the worst that happened is we accidentally put it in.
Noted, thank you.
So, yeah, sounds good.
yeah. There you go. It's in BB4A.
Great. You guys got that? 4A. Yep. And then we'd like to trip a niskin here after.
I'm gonna go poke it for a couple times. It's a nice picture.
The view of all the crabs hanging on the corals in the distance is interesting too. So we've taken one, so this would be Niskin two.
Two. Okay.
Yeah. And we're at waypoint 3, which is probably worth marking.
Put the
Yeah,
flag
we're
in.
going for two.
Yeah, but just look at the flag, that's all.
Alright. Retract in.
slots in the face of the hood so you can watch it as it goes down.
Maybe. There you go. Two is fired.
I'm just gonna see if we can work on this. Yep, both are dirt. So if you, right now, those are the open ones, they're important. You
It'll
can
be
see
in
what I'm
front.
doing.
Nobody, the flag trips around the,
Well, here's how, no, there it is for one. That's
yeah.
fine.
Right, two is stripped.
Okay, so NIST and 2, everybody got that? Thank you.
Okay,
So
fortune.
can you just kind of zoom in on the background, the back part of that rock where all the corals stick up and, or maybe they're just hydroids with crabs on them, which is all interesting. It's kind of like a wall. Those are hydroids, probably?
Yeah, that's something.
Okay. Just checking. It's quite something with the crab ornaments. And these are the some of the hairy crabs. Might be one of the ones we were after earlier. Can we zoom in on the claws at all?
We'd have to get closer.
We'd have to get closer, okay.
We did find
Because we don't know if anything's in it yet. The section's still like,
no, then,
well, I haven't seen anything. Well, we had the end of it plugged, so they can't go out.
Maybe you can collect one. I'm not sure if it's like can you collect one?
Sure.
A pizza?
Yeah, right.
Because I'm not sure.
There's what?
Definitely skip like that. I shall call it deal.
Trash, crabs, fish.
I gotta take pictures of the deal.
So this is the arms of this one. I saw that yesterday. I'm wondering. I don't know if we have this. Greg said they're spines. To me it looks like there's bacteria all over the arms. They look almost fuzzy, don't they?
What all we have left is this. Could you try increasing the ISO? We're just having fun. Because if you see it's like right on the edge of them.
you took the picture already?
Yeah. It's in there now.
So,
Do we have a way to collect that crab?
how many long tallest?
Yeah.
How do you think we could capture that crab?
The one for the garbs?
Yeah.
Yeah.
If, do we have any, we don't have any sort of net or anything right now yet? We could try getting them stuck in the end of the suction and shooting in the box.
We already have 2 crabs stuck in the suction.
Well, if we, he might be big enough that we can just trap him in the
Oh.
entrance of it and then turn it off.
And then put them in the box. Yeah.
Because I don't think we'll be quick. Like, if we do manage to grab them, we'll just crush them.
You wanna go for it?
Sure.
Alright. The mirror position. Keep your arms still for now.
Yep, I just moved it so when the ram comes up, it won't snap.
The question is, will he come up with all the fuzz still on the claws, or will it come off? So, and after that, Anna would like a snip of what we think are hydroids, but could be corals, sitting on the edge of the rock there. Oh, you want that too? That's the black coral.
Yeah, that's for sure.
I mean, I know we did, Greg said we got one of these yesterday, I think. These cool crabs, but.
I don't think so. Or I think what's done.
I mean, I can try grabbing it with the arm. if that
Yeah,
doesn't
yeah,
work,
maybe
then
just pick it up,
I'll try
if
that
it'll
first,
let
yeah.
you.
Never know.
Put it, it can go in
I think that's a great
probably one of the other bio boxes.
Face.
Oh, nice. Iris was on. I always forget to open it, sit up, close it. Take it.
Sure has big claws compared to the size of the crab.
He's on dust.
So who's here?
We'll just go slow. If he goes under.
And Deshana likes,
So anyone should fight. This is tangled. No, good. I'm gonna look at the master view.
Oh, yeah. Can I steal this, Sonny?
yeah,
Is that a question or?
That was a question. Zoom on his face.
So if we think about this habitat, we're looking at all these animals living on carbonate rocks, and these are autogenic carbonates made by microbes. So we really have microbes to thank for this whole assemblage being here.
yes. Sorry.
And the rocks are amazing because they provide a place to attach for some animals. They provide refuge for other animals. They may provide food. There might be some species that actually can eat the rock, maybe those with the radula. They elevate animals up off the bottom, and so there's more current flow and food particles moving by as well.
I wanna get on this side.
Don't underestimate the carbonate rocks.
No. Oh, good, nice.
Good grab. He wants to swim away. Let's just hope he doesn't drop his arm. No, just put him in, it's fine. We had a good picture of him on the bottom.
Yeah, fortunate.
He's trying to fight you, but unfortunately, not well. Fortunately, not well.
Yeah. I can see the back.
Which bio box do you want?
Anything,
4?
yeah, 4B is fine. I mean, are you worried about the crab eating your coral? We'll put him in another box. That's fine. Bye.
Problem is, this little guy can swim out. So let's try. Oh, right.
You'll have to close it partway before he lets go.
Oh.
Oh, no.
Swim away. There's part of him.
We got a claw?
Yeah,
Okay.
he's right there now.
Yes, please.
Okay. So we would like to, if you can pan over to the right on the rock. Anna would like a snip of the black coral, which is the tall curly thing, and then some of the fan-shaped ones at the edge. So that little thing right there, I think she just wants a piece. And then some of the fan-shaped things that are on the back of this rock with the crabs on them.
Maybe I should just get the chemicals. Yes, it is. Oh, that's the chemicals. Maybe the chemicals smash. That's the chemicals. It's the only difference. They're smashed. It's only this one.
Lisa, every node is an exit. Or escape or shark.
What?
Every node is an egg shark. Or
A case?
shark. Yeah, a case.
On what?
Here. Stop. Crinoid. I can see. Sure.
Yes.
Is it the yellow,
Yes.
yellowish thing?
Yes, eggs. Yeah, it's a shark eggs. This one. Now you can see.
Yep. So what on it is what the shark egg case is attached to? Is that a coral, black coral? Or is that,
The one
The one to the right?
is the curly.
no, I know you want this, but what is this attached to?
I think it's hydroids. That's why.
You think these are hydroids?
Yeah, but I'm not.
We're not
expert.
sure.
Yeah, that's why.
They're a funny growth morphology.
We
It's a
can
nice
leave,
photo of the egg case.
We can leave the eggs there.
Yeah, we will, we will. We're gonna collect the curly coral, but
Yeah,
we're just
that's
taking
it.
a picture of the,
Yeah.
oh,
that's
great
fine.
lighting.
Thank you.
Again, it's one of the functions of the carbonate rock nursery habitat. This case, probably it's a cat shark case. You guys got the picture? Thank you.
Zoom.
Okay, so we just want a little snip of that black coral up there. This
That green
one, yeah.
That's the far away, right?
I mean, if you want, I can go full kind of.
See how far it goes? Might have to run and all of
If we're taking a piece, it should have been covered first.
it.
Yes. So the chat people are asking us to slurp the squid, the small squid that keeps coming up. But we need somebody on board to want it.
I don't know what analysis we can do. Maybe for the museum. But
You can ask. Yeah. Well, apparently the squid biologists want to look at it, but I don't know how they would look at it. All these specimens stay in Chile. They don't go home to the United States necessarily, or to, I shouldn't say home, they don't go with the US scientists or the Italian scientists without special permits.
It is a little different, you get used to them going in the middle.
So we've opened section 5.
Yeah.
Who does?
There was you know why?
Why?
Levi tried the bungees and they were excited.
What do we just do? Likewise. Let's see if I can reach, yeah, maybe.
I could zoom in if we.
Well, I just need you to handle it, just so I can see it. How big?
How big of a piece you want?
10
4
centimeters.
centimeters.
That's. I'll also grab an angle and it might be actually better.
Hmm. Are black corals harder to cut?
Yeah, I know.
Yeah.
If we can't, we leave it.
I don't want to strip the skeleton.
So did you hear, she said if you can't cut it, we leave it.
Yeah, I'm gonna try to cut
Yeah.
the same spot, just a little different place in the jaws.
Yeah, very hard.
Is that in the cutter?
No.
I'm trying to get it right at the base of the cutter.
Now, come up with your shoulder, we touch. I open your jaws a little bit.
I think that was in. Yeah, that's, if it's gonna cut, it would be there.
Okay.
You know what I mean? So there's the tight incision.
Slipped out.
Yep. Yep, all of it. Is that in the cutter?
Yeah.
Oh, crap.
There
Oh,
we
you
go.
got it.
Thank you.
See if I can show it. Rotate it around and bring it up a little bit, up in it. Just to try to do my, get a better view of what it is. You want the manipulator, not
That's as good as we're gonna get it.
Nice.
Okay, they got it.
Try that.
Good lighting.
Let's see if we can get it to focus. That's full zoom, yeah.
Yes.
Lights back up. Gotta have it not stuck in the jaws.
You draw it out. Okay, good. Rotate the draw. Yeah, there you go.
Oh, it's out now.
Yeah, I'll just spin it in like spaghetti.
It's out. It's out of it.
We're just go straight down past it and see if it breaks.
Yeah,
Problem with the black rolls are kind of sticky,
yeah.
so it's not gonna happen. Do you think I can get down flat, just without hitting the other curves?
Come on,
There's
don't
no
know.
sponge in the rock.
I
If
didn't realize
it's
that was-
complicated
Yeah.
for them.
Yeah, you know,
didn't
she's suggesting you just drop it in the bio box with the other coral.
Yeah,
Okay.
because the other core is attached
In
to the stone.
box,
So-
can you reach 4B?
Yeah,
Because maybe it's not cheesy.
I mean, this is.
It's just in case there's a risk of losing this here, it'd be better
I
to,
mean, I won't let go of it, I think I'm gonna lose it.
but just in terms of efficiency, maybe? It's off the edge, yeah.
Yeah.
There, okay, there.
I can see it, thank you.
Oh,
Yeah, right there. Open easy.
there it goes.
There we go.
Yeah.
It's in.
Okay, got it.
It's like yellow, it's still green.
Yellow one, I would say.
I think it's too short.
Come down with it. Right there.
No, I need it forward.
Fauna,
Yeah, it's,
you're here. Gonna talk into that microphone and tell people what you plan to do with the coral.
okay, sounds good.
Hi, everyone. Yes, we collect the corals because we are going to preserve these corals here in Chile, but then part of these corals will be analyzed genetically by Dr. Erin Easton. She is working in the University of Texas in United States, and she's interested in the systematics. For us, it's also important to understand which kind of corals there are here. We also want to understand if there is enough coral to then run genetic population analysis for having an idea of the connectivity along the Chilean margin, Patagonia, and then up to Argentina, Patagonia. This is our ideas, but let's see. So this is what we are going to do.
Well, we're gonna go here and just get, so you're sure they're hydroids?
I asked because I wasn't sure. So I asked and they told me that it's leptodecata, and it's an leptodecata, and it's like that is an I wasn't sure
Okay.
because they have this.
So you don't wanna,
No. I mean, nobody's studying here unless you look your rooms.
who did you ask?
I mean,
Yula? So there's still some debate about whether we want they said it's a hydroid, but maybe we will collect a piece with the crab. If you can get both. It's unusual to see hydroids with their commensal crabs, I think, but I'm not a coral expert, and these aren't corals. So what we're looking at now, we weren't too sure if they were corals or hydroids, but we're told they're hydroids, and they've got Galathea crabs on them. If you, I don't know if there's a way to cut a piece with the crab, or if it's,
We'll try.
or if we just take a whole branch. The crab will probably swim away, but maybe not.
If we go too close to them, they'll jump. But if we cut it too far away, I don't think they'll know the difference.
They won't notice, you know.
If we go close to them, then they'll notice. There's rope. There's rope on the left side.
Go up.
Yes.
What are we doing?
Well, I can't tell. But yes, if it was,
I was just precocious.
it's okay. I'm used to it. Sunny, can I zoom in on that one area?
Yeah, go ahead.
I mean, maybe that crab, the far tip would be good. Who else is on there? Oh, look at all the Caprellid amphipods. Can we zoom on those for a minute?
Less glare.
No hydroid. I want some amphipods in the hydroid though. So
So you
we'll
can
take a
collect-
piece
Yeah.
out here. So these little white creatures that look so strange on the hydroid are Caprellid amphipods. They have a series of legs that are hanging on to the hydroid, and then they have some pretty big claws up front. I'm not sure if we can zoom in on the Caprellids enough for people to see the structure, but
No, it's maxed. You focus on this one, retaining the roots.
Yeah.
I'll focus down. What do you want to do? Doing the birds or focus? You're doing the iris. No, you're good. Did you want to focus on the near stem?
White stuff. That makes sense.
I mean, I wonder if they're eating the hydroids or if they're eating other things out of the water. It's hard to tell, but maybe they're eating the hydroids. Again, this is an example when you have one upright species, whether it's a hydroid or a coral or a bryozoan, there's almost always other animal species living on them, sometimes in an obligate way, sometimes facultatively, meaning they might be there or they might not. But definitely anything that disturbs the big species inevitably will create havoc for a whole host of other things that live on and with it. And it's one of the reasons that taxa like these, the corals and sponges, and I'm not sure, but maybe the hydroids are considered vulnerable marine ecosystem indicators in the fisheries world, and they end up getting protected from fishing. Okay. I think people have had a chance to look at the caprellids. Thank you.
So you wanna try to
I'd like to snip the end and the crab, try to get the crab too. But if you think it's just gonna swim away, we had a crab request, so we could suction it or
making
just
that
try.
crab is small enough to suction.
Well, if the hose is already blocked, then
Yeah.
no, but otherwise, yeah.
But if you can cut a piece off and just put the whole thing in a bio box, it can go into either side of 4. It's interesting because I don't see any tubeworms here. Suggests there's really not much methane. But
Are you on the ram or portrait now? Could you get the bio-box? Is it possible that the bio-box ready, or is it just gonna be in the way? Not like open necessarily, just like,
Portrait all of it.
at least, cause that gets us a lot closer to still then. Cause you might not be able to just rack the camera in. And, yeah, when we're ready, yeah, and then just.
Which Microbes?
What do we have?
4, just, it can go in with all the other stuff. 4, there's not too much. Either side.
The
Claw.
claw.
Only.
Maybe 140.
Let's see if it's in here.
the one, yeah. Well, it's the far one. Might get.
Oh, whatever one has the crab.
Once we try to get the crab as well.
What, to smash the crab or
No,
cook?
no,
Yeah,
they want.
I understand that. I can't see what's happening. Let me do it right now. Can you zoom out, please?
It's probably okay to pull the whole hydro, oh, there goes the crab. Right. What happens if, do you think if we pulled the hydroid out from the base, that the crabs would all swim off? Or we could get them? Yep, never mind. We might get, no.
Those two hydroids tangled together.
Okay, and there's a shark egg case on there, so let's leave it. All right, we'll abort this mission. And
You don't wanna-
we can try suctioning the crabs if you see those same crabs, but
So,
So do you wanna collect this?
I don't, oh, you've got it, I see. Yeah, go ahead. Let's
Okay.
put it in the bio box. I didn't realize you still had it. At least we left the egg case. So that's an interesting observation. The egg cases were on hydroids as well as corals.
I just have to smash it over. Oh, it's gonna go away anyway.
Oh, no.
Grab it, grab it, grab it.
Grab it.
No, I
Yeah,
can't.
yeah, grab, 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 grab.
Hmm.
The porch is not very far up right now, is it? Yeah, I can't reach that far on the porch when it's not porched up.
Okay.
That's,
It's stuck by your base right there, you might go look at it.
no, I won't, I don't
Yeah.
know.
Alright, it's gone now. Okay, I got it. Alright, that's on the board.
It's okay. I think we just leave it. Oh, you got it?
Yeah. Good
Okay,
job, man.
great.
Jam the arm in there fast.
Put the entire arm in first before you open it.
Do it.
Put the entire jaw in
Yeah.
before.
Spider. No, close up. Quick. Yeah. Oh, it's too fast.
Half in. Half out. You wanna just grab this piece?
No.
Huh?
Just, because if we're in a better, like, if you have the current, if you're flying around, you might be able to, like, get it to push in with the water. But right now, the current is gonna be just.
So should we just leave it for now?
Yeah.
Okay. All right, why don't we head towards the next waypoint? We're looking for bacterial mats. Want a sediment scoop next to the mat. We want cores in the mat. We'll see if we can find some. I think this feature that we've been sampling is really interesting. It's like a late successional seep
Push the camera up.
microhabitat because there's no tubeworms here, if you notice. The corals and hydroids may or may not have symbionts that can use methane. That red type, if it's Swiftia, someone reported they do have sulfide oxidizing symbionts, which is interesting for a coral. The crabs have lots of spines, so they could be farming bacteria. They could use methane. So there's lots of possible methane use here, but probably whatever it is, they're getting it out of the water, not from below. I haven't seen any bubbles, and Scott's not here, but I'm pretty sure there's no methane reading here, is there? Yeah. Hachi's got the screen in front of her, and we're not seeing any methane at this site. But there are tubeworms right around the edges, so there must be something feeding them. I didn't realize they were right under this, at the edge of this mound. But just on one side.
Yeah, I'm getting it on.
I just wanted to say, because you asked me to.
Thank you.
Just getting the push course out of it.
So in the chat, there's a request for a recommendation for deep sea books. There's quite a few called The Deep Sea. There's a great one by Tony Koslow that's written really for the public, but has loads of scientific information in it. I think it's called The Deep or The Deep Sea. And we did want to suction that very small squid that was hanging around, if you do see one. But I haven't in a while. Another cat shark. Erin, yell if you see anything that looks horrible, please. Oh, that's not a cat shark, is it? No. So there was a question in the chat about whether the carbonates would get covered with ferromanganese, but typically they form between about 500 and 2,500 meters, the ferromanganese crest, so I wouldn't expect up here we'd see that. You need some, I don't know, haven't seen any manganese covered structures here at all. Those are the black, what I thought were black corals when we first arrived. Do you want us to push your background course here or wait until we get mapped? Wait. Yeah, okay. Are you good with that, Paula, or?
Yeah, I'm good with that. The more correlation with passion data, the better.
Here we have 3 heteropolypus. Formerly Anthomastus. Maybe more than 3. Looks like there's some small ones there.
What is our mapping resolution?
You'll have to ask the mappers.
This one,
Yeah.
the M3, that's about a half-meter resolution.
Oh, wow.
Or it's not a map, it's exactly half-meter. Exactly. That's what that one's gridded at, but yeah, we could go a little bit finer, for some of the other patches. Throw that up there. We're gonna keep screwing along this for line.
Before.
So just set it up next to
Yeah.
four then.
Well, yeah.
And I'll just stop it, I suppose.
Stop before that line. Yeah.
What is our distance to the next waypoint?
Just do one more. It is 150 meters.
Okay, thank you. So Erin, do you know how long it takes to form these carbonate mounds? Or how old they would be, the ones we've been visiting?
I'm not sure off the top of my head, but I can phone a friend who's on this ship right now who can probably give me an answer.
Okay. I mean, in terms of dissolution, basically the animal burrows contribute. Animals can contribute to dissolution of carbonate, lowering pH of the ocean can contribute to dissolution, but they last a long time for the most part over geologic time. So I know there are carbonate mounds at seeps that are thousands to tens of thousands of years old. But I don't know how old these are. A lot of times they're formed within the sediment and then exposed later on by currents that erode away the sediments on top. Is that a piece of line?
Yeah, maybe.
The burrows that we're seeing here in the sediment are probably made by either burrowing shrimp or echurans, another invertebrate worm. Chances are they're not being made by fish. We're not really seeing a lot of fish right here. Sometimes the same animal will make a burrow, feed, and then move over and make another, and so you can find clusters of them made by a single organism. And they have different shapes, and chances are that different species are making burrows with the different shapes.
Yeah, I got that bit. Just starting to move now.
Thank you very much.
All right.
So we're still at the site we call El Quisco. We're at the getting towards the northern end of it. I think we've all been really surprised at how many seeps are here and how big and productive and diverse they are. I think the original records were very few. So this is a crinoid on the seafloor. I don't know if people want to collect it. It's beautiful, worth a photograph for sure. Greg's lab studies echinoderms, including crinoids. Okay, he says that's a crinoid we haven't seen. So I think we'll probably want to collect this. Do you think you can pick it up in the manipulator and put it in the bio box? They swim.
Yeah, we'll have to try to grab it before it swims away. One way to find out.
We can zoom in and get a good close-up of it. Beautiful. Looks like it's getting ready to take off. Crinoids are really good swimmers. For those of you who've seen them swim, it's a beautiful thing.
Yeah, we gotta record that. That's all.
Our suction sampler is probably has a couple of crabs clogging it right now. We're not really sure. So the manipulator is stealthily approaching the crinoid, hoping not to disturb it enough that it would swim away. Crinoid versus human.
Greg is suggesting a slur.
Suggesting what?
A slur?
Yeah, but the suction is potentially blocked.
Compromised.
It's gonna land. Don't wanna chase it.
Can you chase it into the bio box?
You could maybe fly it in, yeah. You can try that.
Okay.
So people are getting a good look at how crinoids swim. Where is it?
I'm gonna rack it on your camera so you can see better. Or rack it, I mean. Yeah. It's below you.
Yeah.
Oh, now we're on this.
Mm.
That's it.
I mean, we could try suctioning it into the front end, and then if you can reach it, and then letting it fall out into a mailbox, if it would, but do that without swimming away. Do we know where the crinoid is now?
We'll just keep marking down right where we're, hopefully it's maybe in front of us. Okay, it's heavy vessel left.
Do you wanna
Yeah,
stop it here and you wanna let it go, like, just so it's ahead of you a little bit? Or catch up or no? Doesn't matter. Great, Jarvis.
Yeah, go ahead.
Would you mind pausing the move? I'm gonna leave the waypoint, but stop here.
Yeah, copy, mate. I'm already stopped.
Wow, thanks.
Just call me Rain Man McCann.
I have no problem calling you that.
Okay, we may have to leave that crinoid behind and keep on going. Good day for crinoids. Gets to live. And we got to see it swim.
Bridge, Robbie. That's us moving in.
Copy that.
So you can see the nice 3D map that was made and the mound that we're heading for that seems to be in some sort of depression. That should be interesting. As we traverse over the mud floor, it looks like there's not much down there but shrimp, and occasionally other things. But the truth is that there's tens of thousands and probably millions of very tiny invertebrates inside the sediment, polychaete worms and crustaceans, probably at densities of a few thousand per square meter or more would be typical. And the only manifestation that we end up seeing is like a little darker tube openings and a little might look like pebbles at the sediment surface or little holes at the sediment surface. But deep beneath it is lots and lots of animals in what we would call this the background sediment or the normal continental slope sediment. And the shrimp are probably eating them, among other things. Shrimp are scavengers and carnivores. So the shrimp aren't just sitting here to sit, they're probably foraging, waiting for some motion in the sediment. There's another skate. Oh, that might be a ray. I don't know. It's a different one. It's not a round nose one. Starting to see, I would say, the presence of the squid in the ray maybe tell us we're getting close, along with our wonderful tracker. What do you call this map?
Is the map screen.
Navigation.
Do it.
Nav screen.
Nav
navigation.
screen, our navigation screen shows really well, both the sonar, our waypoints, the Sebastian, and the vessel, right? The Falcor too. Things have come a long way from the very first ROVs in terms of what you have access to in here. Also, we little containers with, a couple of screens up in front, and everybody was crowded in. This is a nice, spacious area.
It's just right for your sonar.
Within the Dipterus family, the skate.
Say it again, which family?
Dipterus?
Dipterus. Okay. So we're about 80 or 90 meters off of Waypoint 4, which, according to the map made, is in a slight depression but then looks like a large mound, quite a few meters high. So we'll see what we find there. The dive before us had what they call the M3 is a mapper. Do you know what kind of mapping it is done by the M3?
It's kind of a pseudo multi-beam. It's more of a forward-facing sonar, but it's a Kongsberg M3 that we've been able to pull soundings out of. And Jeff Beeson has been working with Dave Caress to actually pull out amplitude data to get some of the backscatter from this. But he hasn't been able to get any today. We've just got the bathymetry for now. He needs to sleep.
Yes, Jeff hasn't had much sleep. So the Kongsberg M3 was on with the BBC cameras on the previous dive, which occurred during the nighttime.
I'm trying to decide who it is by the smell, and I can't figure it out. No, it's pretty.
What?
I was hoping I could figure out who it was already, but I don't know what does that smell like?
I was like,
Is that-
excuse me?
That's true. What do you recommend for dinner?
I highly recommend the mushroom soup. Highly, highly, highly.
That is good.
What?
Looks like there might be a few tubeworms here.
It's in the shadow.
It is weirder things.
People are starting to pick up some seepage.
It's in the shadow. The mound come out.
That would be the mound.
It's
Yeah.
the black in the shadow. This here is not the
Okay.
shadow. The Mount Arizona.
So we're there already.
We're almost there.
Yeah, 10 meters of us.
It says your cup.
Ben.
Good morning.
Second time I've heard that sound.
Buenos dias. Buenos dias. Good morning.
So we've just arrived and we're changing the watch right now. The pilot watch, and then we'll be changing the scientist watch, and we've just arrived at a really, what I think is a very large mound.
Minutes of freedom.
Who do you think is coming to replace Sunny?
We've got, that should be accurate, there's probably 2 crabs stuck in the house.
Who's
That
on
should
your
be
watch?
20, right?
Scott?
I don't know what's coming first.
Okay.
So maybe it's not so high. I don't know.
Not necessarily one that wants to escape the box, let it fly out.
I thought it would be a pretty high feature. But there we go. There's tubeworms all the way down the back end of it. Yeah.
I think so, yeah, so good luck.
Look, it's not a factor. I'm not worried since Juliana's beside me.
That's right.
That's right.
Waterarm's got a new cable, so. Let me fire this into one.
Roger, thank you. Put my flying goggles on.
I like your pants. That's like a company issue.
I was hoping we'd come see some bacterial mats on the way over that we could core.
There's time.
We'll come back around over it. Yeah.
Well, look who it is.
Morning.
This guy.
This guy.
Yesterday.
How's it going?
But
I did this to them.
they changed out the
I know.
heel on the heat
We just arrived at Waypoint
Yeah.
4. Haven't looked at it yet, but we're on, there's a mound right
So when
behind
you
us.
first came on, back to the kind of top on the heel.
I'm just gonna grab this as an
And then we
extra.
went over a full, couple hours with full checks on it.
Yeah, there's not much. The biggest problem is that this first section might have got some crabs in it, because we never saw them.
So we're just at
So
location number 4.
we could try.
So I just overshot it. Between like around and fly towards it.
Lots of biomax.
You guys do whatever you want. The suction hose, it's probably long over time. We tried to suction
They all
them
want
into
bacteria,
5.
so
But they didn't
follow
have a sort of
the
time for them.
numbers.
So in the
Pores.
so you can't really use it for suction.
Okay.
They definitely want two background, a couple of background tours, but nobody's wanted to take them until they found the bacterial ones.
Oh man, all the guns come.
So yeah, there's 9. If you can't get 9, for sure we need 7 good ones. So,
She liked it.
Yep.
and then 2
So that
background
was the Mound right there.
tours here and 5 for Elohio, not in the bacterial mat, but near the bacterial mat.
You might wanna knock those out. Okay.
But we haven't seen it. We've seen one very small bacterial mat that's about it.
Okay.
Sorry, I broke that.
Dive, so.
He's that guy.
Alright, boys, come back too.
Tell,
Come back whenever you want. You can come back anytime you want. Just because I like people. Start with all the hard push towards me. That was good.
Good afternoon, everybody, from off the coast of Chile. Afternoon here. We've just had a watch change. Pilots and science team have switched out. So we're going to try and get up to speed quickly and keep on sampling. We are on our 4th sampling dive here off El Quisco, looking at some remarkable seeps. I'm Greg Rouse from Scripps Oceanography. I've got a science team of Anna Adama from Norwegian. Where's that? Tromsø? Okay. And Fernanda and Peter joining me for logging. And Scott, as ever, is here watching the SAGE sensor, telling us all about what the methane readings are. And I've got Scott and Ben doing the hard work of running the ROV. Hi, Ben. Hi, Scott.
Good morning.
And Julianne's running our
Or pass, or 1 minute pass.
social media communication with all of you.
This is the mound.
They just found? They're just circling
Passed
around it?
over and came
Okay.
Came back to it.
So-
A little high.
Sounds like there might have been trouble with the crab in the suction hose.
Yeah.
I hope we
We're
can
gonna try
figure
to
that out.
Cycle that.
Yeah. Otherwise, our main goal will be try and find some bacteria mats to do some more coring. But they haven't seen much mats so far today. How long have you been based in Norway, Anna?
I started last year, January, so it's almost 1 year now.
observation and more using a systematic observation finding approach to more scientific data-driven approach that actually help to prioritize area protection and then plan design these RFPs.
Great. And here, the information we may be gathering off Chile may give us some guidance about making this remarkable place an MPA. Can we zoom in on that? I'm not quite sure what it is. I think it's a giant sea mouse. Yeah. Whoa. Okay. I came on at the right time. So that's a very large polychaete scale worm. You can see the overlapping scales on its body. Now I really hope the slurp's working. Can we get some lasers on that?
Let's see if we can get it unclogged. Lasers on or off?
On, please.
It's on.
There they are. So that worm is about 10 centimeters long. And
You're talking about the pink thing.
the pink thing, yeah.
Yeah, if it doesn't work, we don't. Can I get
So there's a group of scale worms that grow quite large in the deep sea, Admantelini, and I think that's what this is. We had a crab, though, maybe get clogged up in the hose earlier today, so we're going to see if we can clear it up.
Yes.
So we're just running the slope system to see if anything will come through into the chamber.
How many crabs might be stuck in the
At least one, maybe two. Don't think either of them went through. Yeah, they did two into chamber 5. I don't see anybody in there.
It's moving? Suction?
It is sucking, yes.
So we could index to 6, and hopefully this will work.
Okay, there's 6.
A long ways.
You want to move? Yeah.
This one might try and swim away, Ben, just so you know.
Thank you. I think it's further now.
Still can't reach?
No, it's further now. Actually. I didn't want to stretch it.
So we keep seeing new things, even after 4 days of looking. We just saw a feather star that got away. And now this worm is also something we haven't seen. So we'll keep on sampling for the rest of the day to try and document this site. We've pretty much caught all of those various crabs you can see there. This looks like 4 or 5 species we've brought up. Of squat lobster, I mean. There. So scale worms are annelids. They're related to earthworms, which are all segmented. You can't see the segments because soft appendages called cirri have expanded to form those scales. And mostly we get a lot of different scale worms at seeps and vents. Nothing I've ever seen so large, though. Seeing a lot of scales come through. I think there is a crab caught in the hose.
Maybe on and off, Scott.
Yeah, I've been doing that.
So the pilots keep trying to clear the hose. This might still stuff coming through. There are the crabs.
There it is, multiple.
And more.
What does a crab have?
One
That was
crab.
it, right? That's the slug.
Both crabs?
Yep, slug came through, there's a slug. Isn't that it?
Yeah.
Is it?
Yeah.
The worm is there? Oh, great. Okay. Happy day.
Happy day.
Well done.
Very
Thanks,
large
guys.
Nice.
Well, worms make me happy.
Swimming
Not
around
everybody,
right
though. Oh, look at it. It's a beautiful animal. Okay. Thanks, guys. That was very cool.
Anything else here?
I don't think so. I think we better look for some bacteria. There's a lot of demand for
Wait.
push cores today.
You gonna stow it?
Okay, I'll just okay, go ahead and retract.
So you can hold on to it?
Yeah.
You didn't see a coral? Nothing of interest? I think we could just head to number 5, please. And we'll be looking for a big patch of bacteria, and then we'll settle for a push core.
Yeah, I should start it now.
Hmm?
He wants to do push course, I should start it now. And you could fly with George and ram out of the post contact one.
Maybe index the jar to stop the worm swimming out. Thanks.
It's got it.
Great.
Alright, I'm explaining this to our.
Okay, everybody stand by for a second. I got 2 people in my ear.
Yeah, 7's good.
Okay, so. Are you ready for arm then?
Yeah, I got this.
There you go.
So if you watched the dive yesterday, we found an amazing tubeworm mound that had a lot of the Congrio fish using it as a place to hang out. And we haven't seen that at any of the other mounds. It's quite remarkable.
Okay.
And those fish are commercially fished and consumed. Have a look back at yesterday's video early on if you want to refresh. So we're just continuing north of that bush. We were a further kilometre south on our initial dives. And we're just trying to find more, explore more on this last dive here before we head for some canyon exploration. Is that our last waypoint?
Yes.
Oh, okay. We're on the lookout for microbial mats.
Bridge, updated position.
Harley Bridge, up to the waypoint.
So this is pretty
Morning,
inactive
Eric.
looking area.
Morning, Ben.
That was from Sophie.
Sorry, what's that?
That came from Sophie.
Ah, well, good morning, Soph.
That last one.
Coral. We can keep going.
Pretty flat, so.
We're not seeing much on the sonar, but some mapping that was done earlier does suggest something's coming up. Humboldt squid up ahead of us. well-known predators along the eastern Pacific, especially near oxygen minimum zones. Been moving north into North American waters. I'm not sure of their original distribution down here in the southeast Pacific, but they're clearly here now. Okay, bacteria. That's good.
Which one?
Fish just lost out the battle there, I think. We do need to get a bunch of cores, but we'll see if this is big enough. And of course, we've got the trouble with rock. Yeah, it's pretty rocky here. There's quite a few squid. Maybe using us as... Maybe we'll keep going, Scott. Yeah, there's maybe some others coming up. Let's have a closer look. Oh, you see another one?
Move on to the next one?
Sorry?
Move on to the next one?
Is it easy to come back if we... Yeah, okay. We'll have a look at the next one. We need to try and get 9. It's the trick. We might use both. I guess might turn out. Yeah, they're not very big. Okay, let's go back to the previous one, and we'll try and put some in that.
Just one a little bit forward. Towards. See it?
Yeah, you wanted to go back to this one?
Yeah, we'll do them in two sets if we can. If we could get 5 in this, would be good. Peter, does this look like a good mat for you?
Yeah, and one thing we can do to try to make the coring a little bit easier is we can go to the exterior of the mats and hopefully we can get a little bit
Okay. But still matte. You want matte?
Yes, please.
Just the edge of the mat. Okay. Those are beautiful heterocarpus shrimp, the large ones with the long trailing antennae. Might get a zoom in and have a look at it first. I don't see any obvious rocks, so hopefully we'll be successful. So this mat is sulfur oxidizing giant bacteria. And on the surface, but there'd be a whole consortium of other microbes in the core that we might hopefully do. Just seeing if there's any animals on the surface. Yeah, there's some little red feathery... Come down here, thanks, Scott. And just, has it got any more zoom? Yeah, there's some little red feathery appendages coming out, and those are the gills of little worms called amphorettids, which are well known for living in these kind of mats. Okay, let's see if we can do a set of 5 here, Ben. Hopefully it'll work. Mostly around the edge, if you can. Hopefully.
Going for Ben?
That's requested. That's a special request from our friend. I'm gonna turn on lower.
Okay.
We've got about 24 scientists on board this leg, along with a couple of BBC filmmakers. And the BBC crew had a special Sebastian dive last night to use their camera and extra lights to get great shots of the Congrio seep. And hopefully that'll end up in a forthcoming BBC new documentary series, Blue Planet 3. Looked pretty amazing, the footage that we could see. And then they'll be along for the whole of this leg. We're heading south after this. We'll end up in a few weeks after visiting more seeps and canyons off the Concepcion region and switch out with another team who'll continue exploring seeps and canyons and vents all the way down to Patagonia. Yeah. Peter, is that good? Where Ben is? Yeah. Okay, thanks, Ben. As long as it's got some mat underneath it, but along the edge would be ideal. We've had a lot of trouble with the coring because we want the bacterial mats, but the bacterial mats tend to be associated with methane seepage, and that usually has resulted in the formation of carbonates, rocks, and that stops the cores going in. So Peter's asked us to go on the edge, and hopefully there won't be too much.
It's as far as it'll go.
Okay.
Leave it?
Yeah, that's good.
They'll be okay if they come out? Okay, let's keep going.
Okay, relax a little bit. No, it's fine. I'm gonna open it up.
Do you want to just lay them all out?
Yeah.
Okay. We have microbiologists aboard who are really interested in the contents of these cores, but also animal biologists who'll be sieving the sediment. They bring it up and they extrude, push the core down, water comes out, and then you end up with the mud coming up to the top. If we pull off that plunger, and then you can put a ring on top of varying depths and slice off the top of the core. So you can then get sections all the way down, and they'll be used for microbiology, animals, or squeezing out the water that's in the sediments and doing geochemistry on it. So lots of different fates for these cores tonight.
You're good. Are you happy with this, Peter?
Yes, and you can even go closer to the edge. At this point, one thing that we're really interested in is the depth profile. We've gotten lots of mat samples, and those are amazing. So, yeah, right on the edge would be perfect.
Here you go, on the edge. That's it. And open up, stop. Ready?
cores are in special little quivers there that hold them tight. That can be especially important coming up. If they've gotten some methane bubbles in there, which is quite common as we've seen bubbling coming out of these sediments, that methane will expand dramatically and can result in the cores blowing out. So we want to keep them. Does mean there's a lot of pieces to put together every night to get these ready to put down again the next day. So, could be an old tubeworm, could be a little stick. Hopefully won't cause too much trouble. Oh, that core's going in well.
That's a sweet spot. That's as far as it goes.
That's amazing. See if we can place one or two of the push cores to the left of where we're currently placing it.
Roger.
You can use the port horn for that.
Again?
We'll use the port horn for that.
Sutkin. Steel. Sutkin.
Can you summarize what we're doing in Spanish? Or Fernando?
In the mat or in the edge, Pete?
On the edge, please. Yeah, right around where the lasers are.
Lo que estamos haciendo ahora es recolectar muestra de sedimento en el manto bacterial que estamos viendo y estamos colocando 5 push core, que son estos tubos de plástico para recolectar muestras, como dice, de sedimento, y están indicando algunos investigadores necesitan posiciones específicas respecto a la presencia de bacterias, y luego esta noche, una vez que Sebastián volverá arriba a onboard en el barco, luego podrán efectivamente analizar la componente micróbica de los sedimentos, y también otros investigadores harán también análisis químicos del sedimento. Hay muchísimos investigadores que están muy interesados en los pushcores, eso en el sedimento, así como de países diferentes. Así tenemos investigadores liderados por Jeff Marlowe de la Universidad de Boston, y aquí tienen sus estudiantes, doctorado e investigadores también de la Universidad Chilena de Coquimbo, y hay también Eulogio Soto, que es nuestro jefe on board del crucero, que efectivamente está muy interesado también en los sedimentos, así como otros colegas chilenos. Así delicadamente y pacientemente estamos cogiendo estas 5 muestras.
Thanks, Anna. So this will be number 5 on the edge. And we might just be able to put a couple into the mat itself. It's pretty good then. Great. We do want a couple in the mat. Do you think that's too crowded, or
No.
can you get a couple in there?
Sure.
Okay, thanks. So can you note hopefully they'll recover well these 5 of Marlowe, and then we'll do 2 that are for Patchy and Gallardo. We'll see how many Ben can get in there. Maybe at least we'll just go for 2. Yes, please. Thanks, Ben. Right in the middle there would be great. Hopefully it'll go in.
How many are we going to put in there?
2, please. We'll just go for 2. Let's see how it goes in.
This is good.
Yes. So push coring is not easy at the best of times, and when we've got a carbonate crust or layer underneath, it makes it even more challenging.
Very shallow.
Yeah. Try one more, and then we'll call it. Thank you.
Greg, can we get a knee skin before we start filling cores?
Okay. Okay, thanks, Ben. Okay, let's call that. We'll get a nice picture, please.
It's 10.
And the loggers make a nice map. And if you could mark 12, 16, 13, 14, 15 for Jeff's group, and 10 and 11 for Pashi / Biota. And could we do this kid, please, Scott? Great. All yours. Fingers crossed. So what we don't want to see is the core falling out. We don't want to see a lot of bubbles disturbing the stratification in the core. Ben's got huge experience in doing this, so we're in good hands. Looks like there's a clam in this call. Haven't seen any. We've seen little clams alive, but none of the adults, which is that kind of side. There are vesicomyid clams known from here. It's one of the first pieces of evidence that there was a seep here. A clam that was named Ostrogena Neruda I, after Pablo Neruda. We've seen a lot of dead shells, but strangely have not found any of the adults. The sycamore clams have their gills full of bacteria that are using the hydrogen sulfide, and they're farming them in their gills. And the gills are actually strikingly white, a bit like these bacteria you can see here when you look at the clam. So this core was in the mat, and obviously there's a crust right underneath it that stopped the core going in very far. But it should be pretty good for the bacterial layers on top.
Dirt.
14 always. 14 always.
Peter, that was disturbed. Is 5 got a contingency in it, or do you want another?
Here. That was disturbed. Do you want another one?
Yes, if you like.
Okay.
No, thanks. No worries, Ben. Can you shake it out?
Yeah.
Okay to redeploy? What do you think? Could you put it there, Ben? Would that be okay? Might be a better chance of getting in. But still have some bacteria. Yeah, maybe a little bit over to the right. A little bit, yep. That's good. Perfect. Pilots are using all the leverage they can from the ROV to try and push that core down. But they've got to do it in a way that keeps it vertical. If they go lateral, it'll just rip the core out. Part of the issue seems to be there's a lot of shell fragments in the core base, and that is helping the core slide out. But here, this one seems to have worked out fine. The fragments are of that vesicomyid shell I was telling you about, Ostrogena Neruda eye. Great. got a whole lot of probably krill, maybe also some mysids around us here. They're probably attracted to our lights, and above the ROV there's probably a cloud of them. Bush growing's a slow and patient business, so bear with us. We'll be off exploring in a few minutes.
I think we already fired one. Yeah, we're gonna filter anyways, so if there's a sediment or a reason, I'll go worried about so much as like.
It's just falling
We're gonna
out.
filter it anyways, so I think it's okay. Yeah, we filter sterilize. I would prefer before if that one fired correctly, but if you want to push it, if you have bottles to spare and you want to push the seconds, and the first doesn't work, I'll use the second instead. That'll be fine. I don't know. Does it? Are you using the other for eating it?
Yeah, then I can use it. But yeah, if I'm closing for you.
Okay. Yeah, one more would be good. Yeah.
You could shake it. Try again, please. Yeah. You could go back in the mat if there's anywhere you think you could reach.
If you can reach like kind of behind 12-inch on the edge of the mat.
This isn't for you. This is for Patchy.
Patchy, when I'm inside.
We'll still try and get you an edge mat before. Would that work?
When we do the one for Aaron.
Yeah, we'll do some more. Hopefully only one more edge one, but if we need to, we'll do two.
What's that, John? Is that for the ready? You know? We'll fix on our new AC. Is that good, Greg?
Yes, thanks. Thanks, Ben.
Just.
Just for makeup, because yesterday, just for makeup.
Filter cleaner. Filter cleaner?
Can we close the seafloor? Because Tata mentioned that we can double to use
Pushing up.
Just sticking there, Ben. Is it just sticking there?
Exercising the wife though? What's going on?
Sorry for the noise, we don't know what it is. Okay, that could be useful. Thanks, Ben. Let's put this one down for biota.
I put Pachi.
Pachi
Pachi.
biota.
Yeah.
Yes. Yeah, true. But we'll try some more.
You were right, Scotty. I know my noises.
Scotty used to be a ship's engineer.
Exactly. My very first power job, I'm standing on the deck with our OB boss, and the engineer comes out with like motor, chain fall, puts it on deck and starts demounting it, and I look at it and I go, I don't know how to do that.
Okay, that one was disturbed, so we might. Yes.
That was viewed. Thanks, Benji.
And then 12. Yeah.
So Peter, do you want to explain what you're going to do with these cores?
Sure. So what we're trying to do is look at the diversity of the microbiology in both the mats, but also the sediment underneath, which is why it's so important to get these intact stratigraphy layers to determine not just the microbial diversity, but also the diversity of the geochemical reactions that might be occurring at mat sites like these. We're also looking at the activity of those microbes. So if you're familiar with the 80-20 rule, where 20% of the people are doing 80% of the work, we know that microbes are also like that. They can enter dormant states and they can enter active states. And so by determining which microbes are in an active state with later incubations, we can identify the keystone members of the microbial communities, which is really important for trying to establish ecosystem functions, and then also useful for things like bioprospecting and other anthropogenic uses for microbes like these. It's a really exciting system to be working with.
Thank you. See, doing your push cause is worthwhile. Looks like Ben's got us another good one. Great, Ben.
So we want to do. Yeah, we want to pick up, move, want to grab the core first. Your wrist and break.
Okay. We maybe can do it here. Yeah. Okay. Is Pachi happy with these two? Because this one was pretty disturbed. But one, number 10 was okay.
One is disturbed, it's fine. It's a little bit disturbed. One, the number 11.
I have two more cores I can take.
Okay, I prefer not disturb.
Okay.
Yeah, because yesterday was too much
Is
disturbed.
it okay if it's on the edge? We're getting better cores
On the edge.
of the map.
Yeah. On the edge, not. Yeah.
There are some places.
Yeah,
where
they will try that.
Still
Yes.
some map, but
If you put, then it's disturbed. The edge is much better.
sorry,
Okay.
Scott, we do want to do an iskin before we another is
Yeah.
the backup.
Greg?
Okay. Yeah.
Greg, that's okay.
And we still have.
In the, in the edge, closer.
Okay.
You still have the one.
Yeah, number 4 will do. As a make sure we've got good sample. Yeah. All right, Ben, sorry to ask you. We'll do two more. Thanks. We can try them, one more for Marlo. Peter, we're doing one more for you, so then you'll have 5. And we'll try and do another one for Patchy.
Ready? He's gonna move forward for a minute.
This, we're trying to do one for Gallardo. Can you share? Or you need to call yourself? We did get this one. That was okay. It's shallow, but it's good. Is that okay for you? Just one? Or you could choose the best Gallardo. I think this one's the surface. Talk. Maybe ask for a healthier or he wants. Because you asked for one. So we'll go for 2 more edges. Thanks, Ben. If we need to move, fine.
You ready?
Yeah, just walk forward.
Well, you want the edge, right?
Yes, please. Still with some white, but if we can get in a little deeper, it's going to be better, of course.
This. How much white?
Trying to see the down. Can you? Yeah. Yeah. Yes.
Yep, that looks about right.
Thank you. Maybe pull that one out and shake it out. I don't think it'll work. Thanks, Ben.
I don't think so. Now I'm gonna put crust on this thing.
Yeah. Or it's shells. I don't know. Shell or crust.
Make this thing. Okay, pulling out.
Is that deep enough? Can you use this? Is it deep enough? No, it's too short. How about Peter? Too short?
It's a little shorter than we'd like, but let me check real quick. Love it.
Real quick.
Real quick.
It's going in.
Okay, zoom in. And see, decision.
Okay, we're going to keep it.
Yeah.
Thank you.
So what were you saying, Grace? Not gonna hold?
Yeah, you got me. Thank you. Proven wrong.
It's a beautiful sound.
It's great for the mat on top. Really worked. I'll put that down for you, Peter.
Okay.
Okay, we've got one more try here. And that's our last one with bacteria. Then we'll just be going for normal background mud.
Okay. Same area?
Let's see. For background, how far away? How far? You don't need it? No? You don't?
Not so far from
It looks like you could use a little taproot. Or
not
just
so
a-
far from the bacterial nuts.
So near to that, but still away.
Not really away. It's,
Yeah,
Sorry,
that's
I
it.
got this is another one. So I'm getting you one here.
Yeah, there it is.
Can we also?
This is too short.
Well, I've got. We're going to do one more now. Try again. Try another one. But do you want? We'll try. Yeah. But do you also want a background? Yes. Today. So you'll have one background and maybe three. Three. of
Yeah,
these.
okay, yes. But is it because I don't know how short it is? Because if I can use it, this is probably
Yes.
I can use.
But you need it. The trouble is it's hard to in the map. You can't go in. Because if we go near the edge of the map, the edge of the map, we can go deeper.
Yes.
So we'll try one more
Yes.
the edge.
Yes.
Are we going to this way?
What about there, Ben? Can you reach that? Thanks.
Bonus points for shrimp?
Huh. No, I want that.
Oh, don't tempt me. I love that. Okay. Is this good, or you wanna?
Yes, that's good. All right, let's hope it goes in.
Ready? Ready. Pushing. Let's put up across underneath this thing so.
Too shallow?
So I'm just gonna put a cap on it.
Okay.
Okay, I'm pulling up, Scott.
Ready?
Easy, easy. I'm gonna put a zoom to the side, if you want it or not. Here you go.
You want to try again? Deeper? We can try.
Yes, no? Nope, not now.
It's funny.
Oh, it's still there.
Keep
Yeah.
it or not? Keep it. Okay,
Keep
thank you.
it?
Yes, please. Ah.
Oh, no.
Oh.
No. You don't want to use this anymore.
No.
Okay, I'll put this back.
So now the sediment fell in. We can't use it at all. Well, if we got another core, there would be mud at the bottom.
Okay.
Do you want to try?
Yes.
The surface would be.
Yes.
Okay. She's happy if you try again. Thanks, Ben.
Okay, I'm not gonna use this core.
Can you use that core?
I can use the core.
Use it.
Okay.
Even though it's got mud at the bottom. You'll be okay. We're out of cores. We've got to use the other 7 somewhere else. All right, let's guess where it might work. Maybe down nearer to us, where this was kind of worked down here, near the edge of that white area there.
Okay.
Thank you.
Somewhere over here. Can
Yeah.
Zoom. Science. Back here.
Yes, please.
Okay. And then one again. Okay, here we go. Go down. Settle. Settle down. Settle. Down. Settle. Decide to push on.
Ready.
Okay, pushing. Again, pick them up. Still going down. That's good because that's.
That's
sticking
great.
up. Or this is the sample. This is sticky mud. Okay, that's all. Let it settle down. Okay, so now I'm gonna start pulling up.
Ready.
It's gonna loosen this a little bit.
So we've been trying to get the cores to go in as deep as possible, so there's the ability to get the different strata. All kinds of different chemistry will be going on,
Okay, coming up.
and microbial community interactions. So the scientists need that to be undisturbed and the ability to slice that as long as they can along the core. So we've had some come up that are pretty short, and
Stuff now.
we're hopeful this one can come up with a reasonable length.
Go. Go to the mud. On. There you go.
Look at that.
That's a view.
Okay. So 7 is for Pashi and Gallardo. If she can. Okay, I'm seeing 5 good ones for you, Peter,
Yeah, we're set.
plus an extra that's messy. And we've got 3. 1 short for Pashi, 1 good one, and 1 disturbed one. Okay.
Nice job, nice job.
The 7 we have left are background, so
Is that one to be?
all right, great job, Ben.
Yeah, no problem.
Let's have a bit of a push-call break and go and look
Yes,
at something
please, yes,
cool.
please.
Sure, everyone wants to see something else.
Look, Ben's wrist heal.
Yeah, especially Ben, after all that work. Okay, Scott's going to take us on. Let's see if we can find. Got a target up ahead. It should be another mound.
Oh, Ben, there's more mat up there.
Mm-mm. No more mat today. That is it for the mat. Yeah.
There's some mat on your port side. Almost there.
So if you've just joined us, we are off the coast of Chile, only 15 kilometers offshore, in 430 meters of water. Water temperature's a bit under 7 Celsius. Oxygen levels pretty good. We've come through the oxygen minimum zone, and we're exploring methane seeps. We have been here for, this is our 4th day. And this is the northernmost part. We've come exploring these seeps off El Quisco.
There might be some bigger mats to your starboard, no?
Yeah, we're looking more for other stuff now.
Yeah. We don't want to see a big mat now. That would be bad.
Avert your eyes.
Some targets on your right.
Yeah, something ahead of us.
Pretty much waypoint 5.
Yep.
Just kind of look around here.
Yeah, thanks.
There's some targets ahead, 10 meters up.
There's a big mat.
It's like we're not collecting rocks today for Lisa. So the bio boxes are for whatever we want. We did see a crinoid or two earlier, so I've got my eyes out for that.
All those shrimp feeding on.
Shoot what's eating the shell.
It's a skull of a fish. Probably
Can you zoom in, Scott?
Yeah.
after the Humboldt squid ate the rest, they've dropped the head.
It is a fish head.
It's a rat tail.
It's still kind of fresh.
Yeah. So that's one of our little benthic friends that we see all over the bottom. And I'd hazard a guess that a Humboldt squid grabbed it. It's pretty fresh.
Okay.
Okay.
Camera straight. I think you're right there. 1, 2. There's much more targets in the bottom.
There's some of the living macrurids, rat tails, that we just saw the head of. Okay, so now we're coming up on this mound.
Bridge updated position.
Update, thank you.
So we've got a big skate off to the port. That's where made all that sediment. Sea whip? What are those? The sea whips of Gorgonia? We haven't seen that. Okay. Let's have a look at the sea whip there, please. It's a kind of Gorgonian that doesn't branch, nicknamed sea whip. We've got two there, and then a black coral in the background. Oh, you already got one? Okay. Okay. Okay.
I think it's on with the red coral.
Which one?
Red coral, okay? You want to look at the red coral?
The black coral behind. We'll look at that. We won't collect this. You already sampled it. But yeah, we'll have a look at that one. There's a whole cluster of shark eggs attached to that black coral, and then barnacles have attached to the egg cases, I think, which they've grown, so the sharks may well have hatched out, so that's just the already hatched cases. But we'll get a close-up and see if there's any developing ones still inside. I think they're opened. They've been there a while. Those are stalked barnacles or gooseneck barnacles. Okay, we don't need this. It's a nice frame grab. Which?
It's barnacles.
You want the barnacles?
Yep.
Oh, okay. Okay. I've got a request to sample the fish cases to get the barnacles. What do you think? A quiver?
Quiver.
Yeah. There's probably, string from the cases wrapped around the coral, but hopefully they'll come off easily enough.
You want to get a little closer?
Yeah, please. More to the right.
A little to the right. To the right or left?
A little to the left. There you go.
always talking smack. Okay. Looks like anything but one. Or one or five. First. So the water is all over here.
Oh, the standard ones like this. So I need the second one.
So here you have a pretty nice view of Sebastian's starboard arm, and then our 8 coral quivers that are loaded on the side of the ROV, and then just next to them is the suction sampling hose that leads up to a carousel of 8 different chambers, so they can rotate and we can separate the samples. We've only used that twice today, so we've got plenty of time to do that. We've used 2 of these coral quivers, so Ben's just opening number 6 for these barnacles. So always have to do things in the right order, have it open before we sample. Otherwise there's no way to get it open, because the arm will have the animal in it. But we have done that before. Then we have to put it down, open the cap, then we can put the animal in. All right, let's hope these, I guess, cat shark eggs come off. Wonder if one mother laid all of those, or not sure. They're all pretty similar looking in age. Whatever one's got a few barnacles on it, Ben. Yeah. Trying to choose which one to take. We don't want to disturb the black coral.
Can you zoom in on the sip? Get you more there.
I got more for you.
Yeah.
We've seen cat sharks pretty much on every dive. Not so far on this watch though. Hopefully we'll see one. So the arm there's got some little silicone fingers that'll hopefully grab hold of the old egg case, and oh, there's a nice worm on the case as well. barnacles have been used, these particular kinds of barnacles are often found on floating debris, and it's good to know their life cycle and growth rates because that can then help age things. This has been involved with things like aircraft crashes where floating debris comes ashore, and the barnacles have given an idea about how long very nice, how long the material's been floating for. These are cat shark egg cases hatched out. There's probably no one at home. We hope there's no one at home because what we're interested in is the barnacles. And they've probably been growing for a while. Can we have a close-up? Sorry, Ben. Just make sure there's no one inside. I think they're open. Yeah. Oh, there's a polychaete inside that one. Okay, thank you.
Slow zoom study. So that's good. Fair enough.
Great.
You're wrong about you, Ben. Thank you.
That was cool.
Thank you, man. Nada.
Dan, is it okay?
And I'm not getting Singapore at the moment, but it's good. Trying to check in my observatory as well. You're not getting any ground faults from it, are you?
No. We've got, photogrammetry trees off, but it shouldn't matter. Yeah.
All right, thank you, Ben. So we just arrived at this mound. We'll have a little bit of a look, and that's our northernmost waypoint that we've planned. So then we'll look, see if there's anything else we can sample here. We might do a bit of slurping. Got to do some more push cores, see if there's anything else to pick up.
Lift off and look around.
Yeah, thanks, Scott. Yeah, let's look a bit at this mound. We've got no more targets, so our other option would be to go back. So this looks like a relatively inactive site. We've got few, if any, tubeworms. There's definitely a lot of carbonate, but not a lot of chemosynthetic organisms here. There's a few coily tubes around, but nothing like what we've seen further south. Now there is a Congrio sitting down there on the bottom. We could have a look at this fella. This is the remarkable fish we saw. Oh, no, we can get a nice picture. Ben's ambitious. Or hungry. This is a fish that's commercially harvested, and so we've been pretty fascinated to see it associated so strongly with the methane seep. If you look at yesterday's footage early on, there were quite a lot of these Congrios associated with the tubeworm bushes, hanging out in the tubeworm bushes, looking very cute. And it's becoming more and more evident that commercially targeted fish are often associated with methane seeps, which are highly productive systems, so that makes a lot of sense. A good source of food for them. And the BBC team had some fun last night photographing and filming these. It's not very happy with us. Okay, thanks, Scott. Yeah. This?
Yes.
Oh, okay. There's a coil. Yeah, can we just see what this is? So Anthomastus, Heteropolypus, contracted. Yeah, so that's an octocoral contracted. The tubes, what are they?
Fish is turd. Congrio?
Yeah, Scott thinks turd, fish poop, could be a good guess. Yeah. But the octocoral's pulled in there. All the polyps are retracted. Okay, we can keep going. But we go and have a look at that rock over there. I think we've got a crinoid here. That would be, this is one that'll swim away. So it's one you need to slurp. It'll try and swim away.
That's fine. Having more fun.
Yeah. We missed one earlier today. So I'm interested which one this is. It's a lovely rock. We'll get a nice image of it with the squat lobsters and the crinoid. Crinoids are echinoderms.
Put that in jar 7.
Yes, that would be perfect. So they're related to sea stars, sea cucumbers, sea urchins, brittle stars.
First. Beauty shots.
Yes, please. This one is a feather star. It holds on with little cirri, so it's hanging onto that hydroid there. Has 10 arms. And then the relatives of these are called sea lilies, and they have a stalk and attached or permanently or can move around. So we've, in my lab, have worked on the evolution of crinoids. And feather stars evolved from a sea lily ancestor. The mouth is uppermost on the right. The cirri, you can see that it uses for holding on, grabbing onto the hydroid. And this one has 10 arms. I'll be interested to see what it's related to further north. We've got a lot of sampling from northern Pacific, but not from down here. All right, thanks. It's likely a swimmer. Could use its arms and try and get away from us.
Which way you going?
Just about to have a watch change with the pilots.
Right now, we're sitting on bottom. We're gonna sort this guy up. We are at the last waypoint. After this, probably backtrack, looking for interesting things.
Copy. 2 bottles left here. Jar.
I saw that. You should do it.
Hello. Good afternoon.
Welcome back.
Yeah.
2 dives. Jar 4?
7. 7.
We went 7? Okay.
Yep, we can, where it is good. And Doug, this one's gonna try and swim this way. So if you come in at it from the, well, obvious direction, it should. It's pretty amazing how agile they can be.
Go for Crinoid?
Yeah.
These like to jump around.
Oh yeah.
But they run out of energy. They swim for a while and then they get tired.
Yes, that's true.
And then once they're tired,
They do get tired. Yeah.
And I'm ready on suction. I'm on it.
Crinoids are very abundant at the reefs of the Indo-West Pacific and can be in beautiful colours, but much less common in cooler areas and the Caribbean even. There's not many, but if you ever snorkeled or dived on Pacific, Western Pacific, there we go. Easy. Didn't swim at all.
Oh, there it is.
There it is. Okay, we caught that one easily
Anything
enough.
else, or have we done suction for a while, or?
We cleared the crabs out.
Ah, they finally came through.
Yeah.
Nice.
Yeah. Took a bit of wiggling, but
I'll bet it did, yeah. They were a bit stubborn.
yeah, so they ended up in box 6, canister
okay.
6, so yeah.
Are we gonna be suctioning anything else, or do you want me to put the hose away?
Actually, why don't we go some suction over the surface of the rock? Just, we'll get the little creatures living on the rock. So we'd need to go to, say, canister 1.
Okay.
Would be good.
Alright, you're in canister 1.
Just anywhere where there's kind of sediment. Thanks, Doug.
Okay.
You want suction?
Sure.
Alright. Here you go. It's sucking.
So as well as all the big creatures, we're trying to document a lot of the littler things that we can't see. So we'll get this surface sediment, sieve it, and look at it under the microscope tonight. It's one of my favorite things to do. This rock has anemones, hydroids, octocorals, bryozoans, sponges, maybe can see a polychaete. Of course, the two macrurid rat tails hanging there on the left. Spot lobsters. There's a whelk on the middle of the right-hand side of the rock. Looks like it's laid some eggs actually in the hydroid. We'll get a zoom in on that so you can see.
Greg, is that a sand dollar next to the suction?
No, that's a tunicate or a sponge. If I get another look at it, I'd say it's a sponge. Yeah, don't worry about the snail, Doug. We'll actually do a video of that because it's laid those eggs. You can see out on the right. That'll be a nice frame grab after we finish. Yeah, that's good. Okay, we love. Thanks, Doug. That's good. And let's have a look at the snail. And I think those are little egg capsules that it's laid there. Just put the suction sample away and then we'll get in and have a look at the snail.
Which jars did we just do?
That was number one.
So they have.
So that's a predatory gastropod, and they make those kind of egg capsules. So each one of those little structures on the right there potentially has one little snail embryo developing in it, and they often like to perch them out on something like a hydroid to be safe from other predators. Sometimes they can make huge masses of these egg like cylinders or big arrays that wash ashore on the beach, if you've ever seen one beach combing. Okay, I think we're good. Sorry, can you come up one? Anna just wants to. Oh, I see. Over here.
Yes.
Okay.
Gold coral?
You've had that?
I don't know if Erin has this one. I'm not sure. Maybe we can cut that bit.
Okay.
Just one
Maybe
branch.
check that
small,
right.
just to be sure.
Okay, we're going to subsample this,
Oh, yes.
guys. Thank you. This one also has eggs of the whelk on it. So this rock's a large carbonate. How and why it's sitting isolated out here is a bit of a mystery. We saw some pretty huge ones yesterday as well. But they're created by microbial activity that's precipitating calcium carbonate out of the water. So this arm has a special pair of cutting blades that will try and just cut off a piece of this colony. Should give plenty for anatomy and DNA, but leave it to continue to grow. Trick is these octocorals have a proteinaceous skeleton. It can be hard to cut through it. But Doug's done a great job. We'll get a nice frame of it. And we got a bonus hydroid too. With eggs. Yeah. So the polyps have retracted, but we can still see, if we get close enough, even the spicules, the little calcareous rods that are used for taxonomy. Octocorals, of course, have 8 tentacles. Can we get any closer? Nice. beautiful. The hydroid there on the left has skeleton shrimp hanging out on it. They're a kind of amphipod crustacean. Very endearing creatures, but they're hanging there by their back legs, waving out in the current, trying to catch plankton. Some of them you can see have a little bulge in the middle, and they're females, and they've got their babies in a brood pouch there. They have little shiny white eye spots. Okay. Perfect. Okay, thanks guys. So we're up here. We don't have any more waypoints, but there was some microbial mats back here. So we might head back south. Or we could lift up. I don't think there's any more north of us. Let's have a bit of a look though. Okay. And then we'll swing back south.
starboard rim in.
So this is the northernmost we've been in this El Quisco Seep area. We've mapped it pretty well with both the ship and then mapping from the ROV, giving us a pretty good idea of just a couple of square kilometres of what is here, and there's been a whole series of mounds, carbonate mounds, some of which have been covered with gigantic tube worms, so a big surprise to us. We had no real expectation of something as spectacular. And we're going to head back south the way we came today. Got a bit more push coring to do. And sloping. See if there's any other creatures we haven't seen so far. We have continued to see new things even today after 4 days of looking. But in the end, we've had maybe 40 hours on the bottom to try and survey this place. So that's not a lot compared to other places that have been really looked at carefully. But I think we're going to have a pretty good idea of documenting the diversity here.
So I'm headed north past Waypoint 5.
Okay, not seeing anything on the sonar.
I agree. A couple things off the port maybe.
We could go swing by those and then see if there's any mats out there. That's kind of what I'm looking for.
It's it's not huge. It's just
I think this is what I'm seeing. It's sonar, these little rocks.
Yeah. Yeah.
Check this left one, and I'll go to the
Oh,
right.
there's some tube worms there and a sea whip. We might do a slope of these tube worm bushes. Yeah, it's
Looks like we're in jar 1 right now. Which jar would you like?
oh, let's go to 2. So, Doug, if you can slope kind of around the tubes of the curly tubes along the edge of the rock there and in amongst the rock.
Section's coming on. Here in this area.
Yeah, that'd be great.
Section's on.
Thanks.
Jar 2.
So here we've got a more active site with these tube worms, the lamellobrachia. tube worms we can see here are what I've called the curly form. Relatively short, and they just can form pretty dense clumps. We've sampled some of these for genetics. We're pretty sure of the genus. It's a group called Lamellobrachia. And there are 2 known species in the eastern Pacific. One that goes all the way to Alaska, down to northern Chile, it's known from. And another one that's only been found in Costa Rica so far, so we're not sure which one this is. But what we're interested in is all the little animals that might be associated with the surface. So Doug's just slurping among the tubes, and where these worms are, there's probably bacterial growth as well that little animals will be grazing on. Okay, can we just pan left? I know we can't slurp over there, but we'll just keep holding the slurp, Doug. Maybe we'll move and go to this kind of tubes here.
Okay.
And there you can see the other morph, the very long tubes draping along the sediment. There's a Congrio negro there, the fish that we've seen a bit of today, but most spectacularly yesterday, living in the large tubeworm mounds. So these mound tubes you can see here, we saw thousands and thousands of thousands of them yesterday and earlier today. Yeah, if you could just run the slope over the tubes in that area, that'd be great.
All right, so let's come on. You're still on the same jar.
Same one's fine. Yep.
Yep.
So it's possible these long forms are another kind of Lamellobrachia, or it could be that they grow their tubes in a different way depending on the sulfide flow, because they need hydrogen sulfide for their bacterial symbionts.
Greg, do we know if the diameter of the tube is indicative of their age?
Good question. There was an interesting experiment done in the Gulf of Mexico with Lamellobrachia where they took down an ROV and with some dye stained the tubes. Those tubes are made of chitin, and then went back a few years later and the blue stain stayed, and the growth of the tube was then past the stain mark. So they grow from the front, and you can see the tubes gradually get wider, and that's indicative that the worm's getting bigger. But what we don't see here is how tiny the tubes get at the base. These worms start out as little babies, much less than a millimeter long. And in the Gulf of Mexico, they ended up assessing that the tubes were centuries old.
Wow.
Because there's a very slow trickle of hydrogen sulfide, but reasonably steady over the years, and these animals could well be quite old as well. But we've seen these two dramatically different growth forms, tightly curled, and then others that are relatively long and don't dramatically increase in diameter as they go along. So the worms might be able to secrete their tubes at different rates. The worm secretes it from special glands on their skin that create chitin, which then, we just got a little crab. So, could be hundreds of years old, could be growing quickly. It all depends on how much sulfide they've got access to. On hydrothermal vents, where there's a lot of sulfide and the vents come and go quickly, in terms of years, the tube worms out there, the famous one, Riftia, that you might have heard of, can grow to well over 1 meter long in less than a year. So there's dramatically different growth rates this group can show.
That's incredible.
Yeah, Riftia is amazing because it's much longer in greater diameter, so they can be up to 7-8 centimeters across and up to 2 meters long. So these worms we're seeing here are maybe a maximum centimeter across. Okay, thank you, Doug. I think we've got plenty to work with there.
Switching off.
Anything else here, or you want me to take off?
We can take off. Thanks. Let's look for some sediment. A bit more push coring to do. Some Humboldt squid in the background there. Had a lot of fun interactions with the squid the last few days. In one of our slurps last night was a large squid beak that had fallen and got slurped up accidentally. And it was covered with lots of little limpets and that were slowly consuming the squid beak. So I had fun taking photos of that, and we'll try and figure out what the limpet was. There's a little cat shark, what we saw the egg cases of earlier.
Okay, thank you.
Throw my message from
Could be a mat over here. Oh no, it's a squid. I saw something. Oh, there's more crinoids.
Matt's over there. Hold on, check
I
this
think
out.
Matt's over there.
Yeah, potentially.
Yeah.
Should I check this out? Nothing on server. That's good. Mound. You're more interested in the mats and the right now?
Yeah.
Okay.
Yeah, we want to sample. Oh, yeah.
What's this?
That could be a lot of clams. Let's have a, oh no, it's tubes. Tube worms and clams.
Hmm? You see him in sonar to my starboard? You want me to go there next?
Oh yeah, let's go have a look. Our sonar's showing us another target. Can we just look at this coral?
Yeah.
Okay. It's familiar to Anna, so we don't need it. There's a little snailfish, a liparid. They are able to live. Very common deep sea diverse group. Known to live down well over 8,000 meters, the deepest dwelling fish in trenches. No vertebrates can live in the bottom of trenches. That, for among animals, the honor goes to sea cucumbers and amphipod crustaceans and worms. Something about the pressure means vertebrates can't get much below 8,000 meters.
Go look at this red guy, go look at the
Oh,
rock.
let's look at the rock. Yeah, the red guy's a large octocoral. Yeah. So this rock has some wonderful basket stars perched on it. Gorgonocephalus. It's a branching arms of a basket star that is filter feeding. There must be a reasonable current here. This will be spectacular to get some images of. A little bit of colour variation, a paler one, and then the deep, more orangey one. Probably the same. So their mouth is underneath, so we can't see their mouth. But they catch their prey with not sticky, sort of spiny little hooks on their arms, and then curl up around the prey and then bring the arm to the mouth. You can see some of those little arms have curled up. They're reacting. We've brought a lot of potential food in their direction. And they have a 5-ray symmetry of sea stars and brittle stars. But each of the 5 arms branches and branches and branches and branches. One in the background's much larger specimen. Yeah, you can almost see the spiny hooks on the arms there. So we've already collected one of these, so we won't bother with any more.
No, my head is changing.
Just trying to get that in focus. There. This is something called Gorgonocephalus. Gorgonocephalus chilensis was described from down in Patagonia. Is this the same? We'll send a specimen to the museum in Santiago, do some DNA sequencing, and we'll figure that out.
Okay, want to keep going?
Yep.
See those two?
Yep.
Hopefully I don't dust the next round. That's
Yeah.
okay. Is it behind me? Curious. I'm just kind of following the sonar right now. Hard returns. The baskets are up there.
Yeah,
We can turn around at any point.
I think we should turn around soon, but it's still interesting things coming up.
Thought he was going to come at me.
Hmm. There's a lot more tubes on the carbonate here. So this is quite an extensive low field of carbonate.
tube that went up.
Yeah, it's just one of the tubes poking up. This will be our final dive here at El Quisco. Tonight we're going to transit a little bit, do some mapping, and dive in a canyon tomorrow. We don't expect to see seeps there. We'll kind of see what the mapping shows us. But canyons are a big part of the 2 legs of this cruise, comparing canyons along the Chilean coast all the way down to Patagonia.
So it'd be a good spot.
Great job, you got updated waypoint.
I think we might I think we can turn around. There was some bacteria back here. So we'll head back
Hold
to-
off on that move, Eric. Might
Sorry,
be
yeah,
back.
sorry, Doug, we're going to turn.
Thank you.
Sounds good.
It's
South of Waypoint 5.
Are we going back to?
south of 4. Oh, between 4 and 5.
Between,
Okay.
Yeah.
so we'll head to 5 and then we're okay. All right. Turn and report back.
Okay, Eric, waypoint's updated this time. We're heading back towards the south.
Heading back south.
So we've got lots of pink octocorals, several different kinds. Lots of carbonate. The white coils are the tube worms. As you can see there, those squiggly white coils. The longer brown squiggles are other tube worms, the same kind potentially. Here comes another basket star. We're just going back the way we came. We want to try and get back to some sediment, do a bit more sampling. But all of this imagery will be useful for documenting the site later on. So Humboldt squids flying by. So we're on the lookout for some bacterial mat. We could put down just here and get a couple of calls.
Okay. Bridge, we're gonna come to a stop here.
Open up.
Thank you.
Ready for porch operations? Is that enough? This is your 1, 2, 3, 4, 5, 6. I think 10 is open too.
Yeah. That's actually number 9. There's nothing in this one, right?
Yep, 9's free. We'll just do 2 here. Thanks,
Okay.
Doug.
Anywhere in particular?
No, just not on a shrimp.
It's not on a shrimp, okay.
Does the shrimp disturb the?
Yeah, they
Top
like
of
the
it?
surface. When they slice, they want to have it as undisturbed as possible. So a shrimp on the way up would totally make a mess. That went in much easier. If you were watching the push coring earlier today, it was quite challenging. So these are for Patchy and Paula.
Alright, so just those 2?
That's it.
You need a map or?
Nice
Good
grab like
pictures?
that.
You good
Yeah,
like that?
that's good.
Okay.
Yeah, thank you. Listing? Listing? To finish, what it says here? Well, okay. He will come in at 3. He will come at 3. He has. If you remember the cores earlier, these are dramatically different. No black. Much longer. It went in much further, twice as far. Which is a sign of much less methane activity here. That was 9 and 6, 6 in the forward.
All right, perfect, thank you.
We'll just hold here for a sec. We're seeing if people want a Niskin.
Alrighty, that's good, no problem.
We'll just see if they do. They don't? Oh, okay.
No Niskin? Okay.
No,
no
let's
Niskin.
go. Let's head past.
We're just north of Waypoint 5 still.
Yeah, let's just go past 5. There was some bacterial mats there. More.
Bridge ROV, we're gonna resume going south.
Oh, okay. Now they want to.
ROV bridge, was that continue mode?
You can do it.
Yeah, you can continue. Alright, continue.
They'd like the Niskins. We just had a request. Thanks. Both? Both bottles?
No, just one.
One bottle. Okay. Just number 5, please.
Alright, here we go.
There you go. 5.
Here you go.
Thank you. Okay.
And now. Passing 1.5.
Yeah, just up ahead, I think, are some mats we sampled here before. Yeah, there's some mat.
Do you want to do push cords in there?
We don't need to push core in the mat, but Olohio is going to come on. He might want to push core next to the mat, I think is his plan. So I'm going to hand over now to our chief scientist, Olohio, and he'll continue the sampling.
Ray and Jarl, will you slow down and stop here?
Thanks, guys.
Suction.
Yeah, suction sample. This
Fish photo bomb.
Hello.
How are you?
Good.
Excellent.
Hello.
Hello. Hola. Hola, hola. Welcome back.
Yeah.
How are you? Good. Okay.
Can we make a close up?
Close up?
A close up. Yeah, with the suctions.
Ooh, suction sampler.
Where we can see
The little guys coming out.
so
Okay.
there are polyps there. So
Telegram coming out. Okay. We bring your camera over for a sec.
I talk with string?
There you go. Which? Let's see, which jar? I think we're gonna go to jar 4 next. Or 3. We have 3. Little index. There we go. We're on jar 3. Alright, so just those little frills are coming out of the white stuff. Let me know when you want me to stop. So those little red frills coming out. You're getting scared of those. Alright, here we go. We're sucking. You're right there. We're sucking. You're pretty small.
3?
Jar 3, yeah.
They duck back down.
I bet you just take the whole thing out. You'll get it. Yeah, that's right where the little red things were. I bet they came out of those little tubes. Yeah. You-
You could dig it in a little bit. We want the apparatus.
Yep.
So dig in a little bit on the mats.
Yep.
You saw the apparatus, but we
I
didn't.
saw the tentacle.
So just push it in a little bit.
A little more.
Push it in more? Okay.
Yeah, okay.
He wants you to dig deeper. There is one way over left. See this red guy way left, Doug? That little guy?
Can you
Yeah.
see this one?
I think they're tubes go deep, so you're gonna have to get them.
Perhaps if you put the suction in a vertical way, maybe.
Oh, vertical.
Yeah, I think
Okay.
we should do that.
Yep, everything's getting vertical orientation. It's gonna jam it in. Here, let's zoom out so you can acquire. We'll go in together. I can zoom in really easy here. Nice. Look at that.
Perfect. Great. In the same container.
Yeah, we're still on 3.
Great.
Yep. Cool. You want more? Is that good?
No, it's enough.
Good. It's enough?
Enough.
All right. I'm going to keep sucking for a minute while you clean it out.
Muy buenas tardes. Les habla Eulogio Soto, jefe científico de la expedición Chile Margin, cañones, filtraciones y chimeneas hidrotermales en el margen de Chile. Expedición realizada a bordo del barco oceanográfico Falcon 2, perteneciente al Schmidt Ocean Institute de Estados Unidos. Expedición en colaboración con la Universidad de Boston a través del Dr. Jeffrey Marlowe, la Universidad de Aveiro de Portugal a través de la participación de Patricia Esquete y a través también de la participación activa de investigadores y estudiantes de la Universidad de Valparaíso, de la Facultad de Ciencias del Mar, de la Escuela de Biología Marina y del Centro de Investigación Costar. En este momento nos encontramos en un sitio que hemos estado describiendo, que hemos estado explorando, que nos ha interesado mucho por su interesante diversidad de organismos bentónicos en torno a un ecosistema en donde también coexisten mantos microbianos, sedimentos reducidos, con poca presencia de oxígeno y también agregaciones de tubos de poliquetos que permiten la presencia de otros animales como corales, hidrozoos y la interesante también presencia de recursos comerciales pesqueros como es el Congrio colorado.
Would you like us to do push cores or keep moving? Onward? Forward? Okay. Let's go. Vamos.
Los objetivos de este crucero van desde lo que es la caracterización del fondo marino, generando mapas batimétricos que permitan detallar la forma, los contornos de este tipo de ecosistemas, así como también el estudio de la microbiología, los aspectos metabólicos, las relaciones quimiosintéticas que tienen estos organismos con las bacterias, cómo se adaptan al vivir en ausencia de oxígeno, pero también favorecidos por la presencia de algunos gases. También otro objetivo muy importante es la caracterización de las características químicas del sedimento y también cómo se distribuye la fauna y responder a preguntas como cómo se distribuyen, qué especies existen y cómo son sus interacciones. Nos encontramos prospectando el área de interés, el cual nos ha mostrado una biodiversidad importante de fauna. También nos hemos encontrado importante presencia de peces, de rayas, de tiburones, lo cual nos ha generado una grata impresión del ecosistema. A veces estos ecosistemas no suelen ser tan diversos o en apariencia a veces lucen empobrecidos, pero nos ha sorprendido poder observar que ofrecen y presentan una diversidad muy interesante con formas de vida que observan con adaptaciones que estamos recién conociendo. Iremos relatando durante estos pocos, bueno, nos quedan un par de horas de inversión. Estamos a 432 metros frente a la localidad de El Quisco, en la zona central de Chile. Estamos visitando diferentes fondos en donde buscamos principalmente estos ecosistemas donde hay mantos bacterianos, pero también nos iremos deteniendo en aquellos lugares donde encontremos rocas y podamos observar fauna bentónica que nos pueda interesar recolectar. Aprovecho de saludar a dos investigadores de la Universidad del Paraíso que han obtenido un importante proyecto de colaboración internacional y que son también investigadores del Centro Costal al que pertenecemos quienes estamos acá en el barco. Por lo tanto, queremos saludar y darle una felicitación muy especial al doctor Mauricio Landaeta, de la Facultad de Ciencias, y al doctor Simone Baldanzi, también de la Facultad de Ciencias, de la Universidad de Valparaíso.
Yeah, we put that, we're right between 5.4 and 5.5, yeah, halfway.
We are in the right direction, so just move forward.
You keep going for it, okay.
Okay.
All right. Headed to waypoint 4.
Are we gonna stop at waypoint 4 and continue?
Stop.
We're gonna stop there, okay?
Every time I think they're gonna come close to me, they get shy and like change directions.
Bueno, también les comento que este tipo de expediciones nos permite la valiosa oportunidad de poder recolectar organismos marinos, los cuales posteriormente serán estudiados, identificados y finalmente depositados en el Museo de Historia Natural de Santiago. Expediciones de estas características son muy importantes para el conocimiento de nuestra biología y de nuestro patrimonio natural. Por lo tanto, mantener colecciones biológicas en buen estado, en buena preservación y en lugares adecuados donde se registran, por ejemplo, cuando encontramos un organismo se anota la profundidad, el lugar, la fecha y posteriormente los antecedentes asociados a la especie misma y es muy relevante en la generación de colecciones que quedan finalmente depositadas en el museo en este caso. Por lo tanto, también estamos cumpliendo con un rol importante que permite generar un patrimonio importante. Son organismos de nuestras aguas, de nuestro mar, del fondo marino de Chile, y es importante mantenerlos en lugares en que son dentro de nuestra jurisdicción. Por lo tanto, también aprovechamos de agradecer esta plataforma científica que nos permite acceder a este material biológico de alta importancia y que también permite posteriormente educar, realizar actividades de difusión que la comunidad, no solamente científica, sino que también los estudiantes y quienes no son científicos puedan ver este tipo de animales en los museos. También el material es muy importante para realizar tesis de pregrado y de posgrado y seguir líneas de investigación como es la taxonomía, la sistemática y el registro del material biológico. De hecho, para la segunda parte de esta expedición se incorporará al grupo de estudio de organismos bentónicos el ya doctor, creo, colaborador y amigo Jorge Pérez Chulteis, curador del Museo de Historia Natural de Santiago. Él participará en la expedición que comienza el 6 de noviembre, que va desde Puerto Montt a Punta Arenas, con otros colegas que también van a apoyar este trabajo, por ejemplo, el doctor Matthew Lee de la Universidad de los Lagos, el doctor Américo Montiel de la Universidad de Magallanes. Estará a cargo de esta expedición la doctora Patricia Esquete y, bueno, se incorporarán otros colegas y científicos de otras líneas y de otras instituciones. You can stop and collect the 5 bushels.
You turn my photos back on and turn them on. Bridge, we're going to be stopping here in sample.
Stopping here.
Okay. Watching out.
Said 5.
We score.
5 left.
We kind of start with 1,
Start with
2, 3, 4, 5.
It's 5. You start with 5. Push them all in.
It's fine.
Okay.
So we put all the line,
Put them in a line.
make a screenshot,
Screenshot.
then
And then we'll pull. Okay. Straight lines there. Sure. You need to be moving cameras? Or you like?
I like a challenge.
from a different field? Is it ever different?
I don't think so. I think it's just
Experience?
Bueno, quienes están viendo en este minuto la transmisión, podemos ver que el brazo mecánico del ROV Sebastian está enterrando unos tubos que se llaman push core. Estos tubos se entierran en el sedimento, en la arena, con el objetivo de poder obtener columnas de sedimento no perturbadas, con el objetivo principalmente de, por una parte, estudiar la macrofauna y la meiofauna, pequeños organismos invertebrados, muchos de ellos microscópicos, que viven en diferentes estratos de la columna de sedimento. Pero también se obtienen estos push core para caracterizar la biogeoquímica del sedimento, que estudiamos ahí en colaboración con la Universidad Católica de Santiago y de Valparaíso, a través de la colaboración con la doctora Marcela Cornejo. Se determina, por ejemplo, el tamaño de grano y una serie de propiedades físicas del sedimento. También se hace determinación de clorofila y pigmentos. También se analiza la concentración de materia orgánica, de carbonatos, y también se realizan estudios de isótopos estables de carbono 13 y de nitrógeno 15 en los sedimentos superficiales de estos ecosistemas. Finalmente, también en colaboración con el doctor Jaime Romero del Instituto Nacional de Nutrición y Tecnología de Alimentos de la Universidad de Chile, él y su grupo realiza una caracterización de las comunidades microbianas que habitan estos sedimentos superficiales. Los sedimentos, una vez que llegan a bordo, son procesados, muchos de ellos son congelados, algunos son preservados con algún reactivo y posteriormente entregados en los laboratorios respectivos para que los expertos realicen la determinación de los parámetros que ya mencionamos. El equipo de la Universidad del Paraíso que se encuentra a bordo del Falcor 2 está formado por el doctor Manuel Castillo, quien realiza el procesamiento de datos oceanográficos a través de el CTD, que es un instrumento que baja hasta la profundidad, contiene sensores de temperatura, salinidad, oxígeno, fluorescencia, radiación. Y él principalmente se preocupa de analizar estos datos y así poder obtener perfiles y analizar todo lo que corresponde a la sonografía física de los lugares que estamos estudiando. También se encuentra a bordo la bióloga marina Fernanda Orrego, del laboratorio de ictioplancton del profesor Mauricio Landeta, quien está a cargo de los lances de redes para el estudio de las comunidades del zooplancton, lances que se realizan principalmente durante la noche. Y bueno, el estudio de organismos bentónicos, así como el estudio de los sedimentos y su biología, a cargo de quien habla, y también con la participación del estudiante de pregrado de biología marina, el señor don Yerko Castillo, que nos apoya también con otras actividades.
Yeah, we'll just reach out. 2, 3, 4, 5.
Thank you.
All right. That.
Now the hard work.
Now the hard work. Mike, tell me what you need, Doug.
Get up to like
Surgeon. Yeah, he's pretty good at these.
Great, thank you, perfect.
Start good.
Can we, you look at that?
Yeah.
How big is the shark? The shark more or less, how big? Maybe 30 centimeters?
That's kind of hard to laser. Yeah, sorry about that. Maybe about 30, yeah.
30. Can we go there?
Yep.
I'm thinking to catch the shark.
You want to catch the shark?
If it's possible.
I would like to. Let's see how fast Doug is on the minute.
Shark fast.
Oh.
Just this one.
No. Go for the couple.
Or maybe this, well, later, but. Wait.
Oh, he let it go.
Wow.
Whoa.
Wow. He was
That was
not
cool.
tasty so much.
You must have tasted.
That was cool.
BBCs. Okay. Here.
BBC.
BBC has a thumbs up. Ooh, wow.
Last bottle.
That was
That was awesome.
that buffalo. As
Just
a
gave him a
real
little
fish.
kiss.
There's no way he wants it.
Did you see that?
Yes.
Oh, oh no, he's sad.
Oh, camera's
The
camera's not working.
no.
But you got it.
I got it.
Thanks.
I tagged it.
That wasn't great. You want me to just turn it off, Dan?
Or equivalent 3 or 7.
See if the ground fault comes back if you do turn it off.
I thought your thumbs up meant you got it.
No, the thumbs up was just that it was cool.
Ah!
If you got it as well, then okay.
With the wrong signal.
Yeah, I mean, we'll still use it if you had it.
It's amazing how this thing happens when the camera falls out.
Wow, sweet.
Alright, what?
Put this on coral cover.
Oh, I don't know.
Six of them, too. Are you just
When I'll go into the DVR.
Perhaps if we collect the tube.
Yeah, grab the tube. Okay. I rained out a little bit, not all the way, because it's due to the. Yeah, it's okay. Rain. It's also a little dusty. Ooh, that shark is playing. A little dusty. Yeah. It's only like a little bit dusty. I could take off the rail on the way out, and then. Sorry, I didn't reach it. Maybe just a little. Maybe put lower. Does that make it so you can see this should clear up. I have some forward wet. No.
But I think 6 is.
Yeah.
Oh. 6 is, there it says.
They say something 6, Doug.
What?
Something 6. He's got something 6.
The eggs.
Eggs are in there?
Yeah,
Yeah.
Gotcha.
The barnacle.
Eggs with the barnacles.
Yeah, so
Hold on back up.
available are 3, 7, 4.
Got it. All right, thank you. Discrepancies. 3.
it's attached on that too there, so grab that too. Not sure what side of the tube's anchored.
Oh, really nice. Yeah.
Good.
Perfect.
Anything else you'd like here, or you'd like to keep moving?
Yeah. Well, look at that. It's a bottom.
Oh, yeah.
We would like to have a look around.
Look around.
Yeah. Come, so, okay, and we follow like.
Yeah.
Oh,
Yep.
forward.
Okay. All right, still in this ram here. spit that for sure. It tastes good. What?
That's what sharks do.
Why? I guess we can.
Can we try? I know it's not very too difficult, maybe. No, forget it.
It's so cute.
It's very fast.
Yeah.
No rocks. Ah, that looks like a rock, but we don't need this dive.
Tubeworms? Sponge?
No, I think it looks like sponges.
Yeah.
Ah, this one.
Spot lobster with big arms? You want me to suck them up?
I think we need this one,
You need that
please.
one?
Yes.
All right, Doug, here's my man.
Oh, well, we can start with.
We're gonna sample the spot lobster first, and then we'll do the sponge. We'll come back to the sponge.
No, this one. No, I'm wrong. Sorry.
Oh, it's the wrong one?
I thought.
We don't
I
want
thought
that
back.
You thought it was a different one?
No, no.
Okay.
We do not want.
Because there is a similar crab, but long arm, but it's not this one.
We got a little excited. I was so ready. I indexed the jar to 4. I was so ready. Now I dusted the sponge real good. And up. Who is leader over here? Hold on.
So,
Give me some, Doug.
yeah, in the sponge, can we make a suction
You want a section here too? Okay.
from the sponge?
Yeah, you want to suck the sponge, okay?
Yeah. Maybe 4 or 8.
We'll get you a little closer, Doug.
Yeah, vale. Yes.
Alright, here we go. You got section. You're on 4. That should clear in just a second. There it goes.
Yep.
You want more?
A little bit more.
A little more? Copy.
Is number 4.
Yeah, drop 4 now. Nice.
Yes.
Is that enough?
It's enough, yeah.
Yeah, I think all the pieces are in them, they feel structured. Alright. You need to take off so it's cleaner. I think I can ram further out too if you want. Yeah, 4. Sponge.
Let's see if we find something interesting.
Okay. Let's keep going.
Keep going.
South? Keep going south. Let's
Yeah.
find something interesting.
Bridge ROV, we're resuming on route south.
Resuming now.
Thank you.
Is there some waypoint in this adaptation or not?
Yeah.
Okay.
So
Okay.
good for Waypoint 3.
Wait.
Oh. Picked something off the ground, isn't he?
Fish, I think. Huh?
So, a little lunch.
Yeah.
Snack. He's faster than me. I was trying to chase him. He's so much faster.
scoop surface.
Sure.
Yeah.
Sure.
We can put in any container here.
Okay. Alright, I'm gonna put out. Camera. We're gonna push out further if you don't allow.
You go.
Let's go. Park the bottom. We got. Looks like 8 zones available there. This is kind of followed by zip tie.
Oh, wow.
Check you up a little. You can try it. We barely got 3 close to zip tie. These are wide open. Cool. Let's see, I have one missing model left. The place that was bioviruses. Headed southbound, back on your track. I stopped waiting for the ship, so we thought we'd stop and grab a bag of tourists. Just head southbound, Nice. Yeah.
Okay. That's pretty full.
It's enough, thank you.
Which box we can put?
Preferably in 2 somewhere, because I think that would be the easiest one to read from all this.
2A and B are both empty.
2A is okay?
Yep.
Okay.
Yep.
Close your eye like that.
Yep.
Towards the top, towards your first Bridge, updated position.
Okay, thank you.
That's cool. Because we need it.
Can we collect? Please. Maybe once a quarter.
Okay. Once I'm on the call,
First we can make it closer. Also, if you take from here,
okay.
it's better.
It's not going to swim away.
Yeah, it's also
But then we'll wait.
yeah.
Maybe.
No, if you take here, we avoid the animals from flying. But it's
Just
maybe
grab the animal. You wanna try, you wanna click underneath? Okay.
Okay, now that's a good thing.
better.
More centimeters.
Okay, yeah, biomass.
Some bioboxes in the better.
Yeah, so, did they tell us? There's a zip tie going like this.
Yeah. We said to use these two, like this one.
Yeah.
Some seed.
Oh, yes.
Your choice.
Great.
This is
Good. Well done.
Big one, it's all fuzzy. Got lasers on them right now.
What do you think should be?
Oh, yeah. I can grab that one.
Yep. Okay. Perhaps not take from the arm, maybe take from here. If you take from the arm, you can block it. You can block it.
Okay, but break it.
Okay, come close.
Premio? Premio? No premio? Parece que ya está
Tip jar?
Let's go watch this thing. Got it. Should we zoom in? Oh, I didn't realize it was all arms. That was not arm. Okay. He's here. Alright. ¿Es el brazo más importante? No, it's todos.
No.
El brazo.
No, before. The whole body is possible.
Okay. On the whole thing.
Grab for dinner. Oh,
If I grab this antenna. ¿Tienes antenna over here? No. It's getting comfortable. Is that all you're doing? No, on the side.
it's gonna hold.
Actually, I'm gonna grab some. No, I'm not going to. Just be delicate. I want to get closer. Can I see the whole one? Oh, yeah, I see. So much fun. Using it in science.
Oh. Use the force, Ben Kenobi. Wow. He's like, nah, I don't wanna.
Any boxes, maybe one, like that.
BB2B is also
Yeah.
open, if that's easier.
I'm gonna put in BB2A. Is it okay?
Yes, yes. No, 2B, because there is a scope in 2A. There is a scope with sediment, so it's better, yeah, BB 2B, please.
And then
Great.
Wow.
and break on everything.
That
This bottle. The last one. One?
We don't need rocks, push car ready. Perhaps one more scoop,
Do we have another scoop? Oh, yeah.
one more in any,
In any bio box?
any, yep.
Do we want to fill any of the coral
No,
corners?
yeah, you try, or you can. Somebody else, can be some mollusk.
Do we have to be up by 6 p.m.?
Sorry?
He has to be on the surface by 6 p.m.
At 6, yeah.